Good morning, everyone. Welcome to this joint Heads of Medicines Agencies, European Medicines Agency, multi-stakeholder workshop on patient registries. On site at EMA today, we still have about 80 people after hopefully a good night in Amsterdam for you. We also have 700 people registered for online participation. And today, in contrast to yesterday, we're also joined by various people following online. So this meeting is being broadcast. My name is Peter Arlott. I head the Data Analytics and Methods Task Force at EMA and co-chair the Big Data Steering Group. And it's my great pleasure to co-chair the second day with Professor Patricia McGettigan, Professor of Clinical Pharmacology and Medical Education at Queen Mary, University of London, also member of the PRAC and member of the Big Data Steering Group. We had very good discussions yesterday, focusing on the value of the EMA qualification procedure on patient registries. I think we now have a very good base to further work together on optimizing the process, thanks to a better understanding of the various stakeholders' expectations, a draft checklist to guide the structure and content of qualification applications, and a list of measures for the development of post-qualification lifecycle processes. Not bad for one afternoon, I would say. All these key elements will be summarized in the workshop report, which will be published on the EMA website in the coming weeks. Today, we're going to focus on unlocking the potential and enabling the use of patient registries for regulatory purposes through enhanced description of data quality, increased data discoverability, and the promotion of data interoperability between data sources. There's a housekeeping slide on the screen now, just to remind that for those who are online, microphones are muted and potentially can be unmuted if you want to intervene during the discussion. If you want to raise a comment, please put your comment in the chat box. As those of you who participated yesterday will have seen, we try to include as many of the questions as we can, although we can't guarantee that everybody will be discussed. I would also ask you to leave your camera switched off if you're online, unless you're actually speaking and taking the floor. If you have connection issues, there's an email address on the slide, and we would encourage you, if you're following on WebEx, to leave the WebEx running during coffee breaks, because then it makes it easier for us, just in case you have connection issues when you try to reconnect. So before moving to slide one, to session one, sorry, we have three opening speeches to give different perspectives, to give some context to the day. And I'd like to start by introducing EMA Cook, Executive Director of the European Medicines Agency. EMA.
Peter, thank you very much, and good morning, everybody. It's a real pleasure to be here. I'm delighted to see so many people in the room and to learn that we also have so many people online. So it shows the intense interest in this area, and it shows the value and also the potential value of patient registries for the optimization of the use of medicines going forward. So it really is a great pleasure for me to open this joint HMA-EMA multi-stakeholder workshop. And I would like to warmly welcome all of you. I know that we have experts from all over, from different areas. We have registry holders, regulatory agencies, health technology assessment bodies, industry, academia, research organizations, patients, healthcare professionals, and of course we have the European Commission as well. So we have a very wide range of stakeholders who are all contributing to this important workshop. So I think if you're here today, you're here because you know that patient registries are very important for the generation of evidence on the benefits and risks of medicines in real life. And this is one of the reasons why back in 2015, we launched a dedicated initiative aiming at exploring ways to expand the use of patient registries and also to promote dialogue between stakeholders, but also to provide better methodological guidance so that we could use these registry-based studies for regulatory purposes. So today we have had EMA cross-committee task force on registries, and this is chaired by Professor Peter Moll, who's here and who you'll be hearing a lot of from later. But this task force organised several multi-stakeholder workshops over the years, and these workshops have been key to shaping our current knowledge in the complete ecosystem of existing registries. The learnings we've got from these have been translated into a guideline on registry-based studies, and this is used today as a reference for the conduct of studies on medicinal products based on registry data. But of course, we're at a very exciting juncture in the pharmaceutical ecosystem, and the digitalization is happening at such a speed that the potential of using these registries even better is really on our doorstep to be realized. And this can help us move towards the vision of big data to improve regulatory decision-making by strengthening the place of data collection and analysis from registries in helping us to optimize the use of medicines. And of course, patient registries have a very important role to play here. They have the capability to collect granular data on patients with a particular disease or condition, and also to give us long-term data on these, on how patients are doing on these medicines. They also provide us with a unique way of monitoring patient safety and effectiveness over time. And of course, this is particularly relevant in the context of rare diseases, where we probably have so little data going into use that we need to supplement it even more by the experience we have from the patient registries. But if we think of the vision of how much more we can do, patient registries have the potential to deliver even more for evidence generation than they are doing today. So it's very, very important that we keep the momentum, that we strengthen interactions among all stakeholders to allow us to continually share knowledge about the use of registries in diverse situations. And this then helps us to understand the evolving data needs and hopefully to foster greater access to registries to better support research, innovation, and of course, optimization of patients for medicines. So I know many of you participated yesterday at the EMA Qualification of Patient Registries Day, and I understand that that was a very successful workshop with concrete actions identified. And we can assure you that these will be followed up over the next months in order to make the process more accessible and valuable. For example, the development of a draft checklist to provide the structure and content of qualification requests, as well as the implementation of practical steps to ensure standards for qualified registries are sustained over time. So today, we will be moving forward in enabling better use of patient registries to address some of the known challenges the challenges that we have been mulling over and trying to address. And I think you will all be aware about the work we've done on the data quality framework, which was adopted by the CHMP last December. By having a data quality framework, it helps us to standardize and define the dimensions and metrics to enhance the description and characterization of the data quality, particularly from real-world data sources. Now, also, the new catalogues of real-world data sources and studies which we will be launching imminently, build on many years of reliance on the NCEP resources database and the EU PASS register. these catalogues will strengthen the discoverability of reliable and relevant registries for research and regulatory activities. We're putting these together into catalogues, and I'm looking forward to seeing many more registries included, so that we can increase the knowledge of the registry's existence and also support design and interpretation of studies. Now, today you will also hear about a novel big data platform which has been developed by a European public-private partnership consisting of experts on hematology, and it's aimed to provide a large data lake and state-of-the-art analytics services through machine learning. And the system intends to speed up research on hematology-related diseases, including blood cancers. But why this is so important for us is the methods applied will enable interoperability between data sources, including registries, and they can help us to drive future changes in evidence generation for medicines regulation. So today's event, very exciting, a perfect opportunity to reflect both in plenary and in the breakout sessions on how these tools can be applied to patient registries and how we can reinforce their effective use for regulatory purposes. So please do provide your views and share ideas on how we can improve and collaborate. And this will really help us to shape the recommendations for future work in the field of evidence generation based on patient registries. So thank you all for your participation today. I'm looking forward to hearing the outcomes and seeing how we can use this very positive momentum to really give us better data to treat patients across Europe and beyond. Thank you very much.
Thank you very much, Emma. I'm going to move straight on now to Professor Peter Moll, Professor at Groningen University Medical Center, member of the Committee on Human Medicinal Products, also working with the Dutch Medicines Evaluation Board. As Emma mentioned, Peter was the co-chair of the EMA Cross-Committee Task Force on Registries, which ran for about 7 years and continues to provide important leadership in this area. Peter.
Thank you, Peter. And so it is a great privilege for me to also be able to give a few opening remarks with that background where I was really at the growth of what we did as EMA and the network, talking about patient registries, especially coming also from the planning of studies in the scientific advice, and currently then in that decision-making role where you have to deal with the data that is coming forward also from these real-world settings. And basically there are 3 areas where we use real-world data or registry-based real-world data in, and that is in the planning and the validity of the studies that are out there, the looking at the clinical context and understanding that better, and also looking at association and investigate also the impact perhaps of drugs. And I'd like to go a little bit in deeper on these topics because, as some of you might know or may not know, I'm also leading the More Europa project, where together with a couple of registry owners and regulators, including Bruno next to me, we're really trying to understand better how to use these data. We will work on also training towards regulators, HTA, and within the network to understand better what the expectations are and where you have to look at the critical parts. And to talk about these different contexts where you can use real-world data, I think it's important to realize that, for instance, these data are also very helpful from registries or can be used very much to inform a better design of a trial. If you look, and we did a study in More Europa with the heart failure registry, looking at the use of a new drug in a population, playing with the exclusion criteria of that trial and seeing that if you work with certain exclusion criteria, you get a more efficient trial because more endpoints happen. And that is something that is very helpful in planning the next trial. So that would be one of the aspects. Obviously, investigating representativeness, what happens if you assume there might be effect modification in certain groups that are underrepresented, you can use this in your analysis. For the clinical context, I think the big hot potato is there. What can we do in terms of external controls? What kind of data do we need? And one of the studies that we're doing is, can you use natural language processing to enrich your data sets and see if there's sufficient information in that? Very difficult, and also part where there is a lot of discussion, and that is definitely not worked out yet. Other aspects that I think are very interesting is obviously the investigation of impact and the association. We know association is not causation. We've seen all where a real-world data analysis without randomization have gone wrong in the past. But I think we're learning with that, and we're experimenting with things like target trial emulation studies that are really trying to mimic the trial that it's sort of to follow on. But I think going further there, if you have the registry, if you have the identification of the patients that are really having the disease, then that is also usually a good platform to identify patients for in trials. So if you blend and real world data and RCT are not necessarily mutually completely exclusive, in principle, there are lots of trial elements that can be integrated with real world data. And then you get to something like a registry based RCT. So you randomized within the registry and you follow the patients up using the normal data collection in practice. And that could be a model that is very promising perhaps for drugs that are already quite well established. So I think concluding, real-world data from registries can be very helpful. It's very important that you see it as a building block. A good understanding of the drug mechanism of action is important before you really start, and some understanding of the data in a tightly controlled setting of the trial. But there, I think after that whole context, and as I described, is the way of real-world data that can help us further. So I think with that, I'd like to give the word back, and thank you, and I am looking very much forward to the discussions of today.
Thank you. Thank you, Peter. It's my great pleasure to introduce Simona Martin, who is project leader at the European Commission Directorate General Joint Research Centre, and is currently based in Ispra, Italy, within the disease prevention unit. Simona, I would just like to add medical doctor by background, hematologist, PhD in stem cell research. Great to hear your perspectives about registries, please.
Thank you very much. And I'd like to thank once again the organizers for the invitation. I'm very pleased to participate in this workshop and now to give you an introduction on an initiative of the Commission's Joint Research Center, which was developed in collaboration with DG Santé, namely the European Platform on Rare Disease Registration, short EURD platform, and to point out to the opportunities for integration with the EMA catalogs. Due to time constraints, I'll have to be very short, and so I will just mention a few points. Next slide, please. So first of all, why a European Platform on Rare Disease Registration? Because approximately 30 million Europeans are affected by a rare disease. In this context, the main objective of the platform is to cope with the enormous fragmentation of information about about these patients in the EU, information that is contained in hundreds of patient registries at local, regional, and national level. So one of the objectives of the platform is to help reaching the critical number of patients for studies and research, building patient cohorts. Another objective of the platform is to provide interoperability to the registries, and this via providing standards for data collection and data exchange, semantic interoperability, fair data, and providing tools for data linkage and data transfer. The EURD platform is already operational since a couple of years. It's online, and that's the link. Next slide, please. As we have heard yesterday and also today in the introductory speeches, the aims of the EU platform are the same as the ones of the EMA catalogs. And one of those is ensuring discoverability of data from participating registries. We know how important it is making registries data searchable and findable by all interested parties. Participating in the EURD platform, as in the EMA catalogs, gives visibility to the registries. And the registries always remain the owners of their data and can decide about the use of this data. Next slide. There are different opportunities that we see for better integration between the EURD platform with the HMA, EMA catalogs. And these are related to the different components of the EURD platform. One of those is the European Directory of Registries, which is itself a catalog of the participating registries containing up to 40 characteristics of these registries. This Directory of Registries could be aligned with a metadata list of the HMA EMA catalog. Another core component of the EURD platform is the Data Elements Repository, which is nothing else than a collection of the data schemes of the registries. This has the potential to enrich the EMA catalogs. Both the EURD platform and the EMA catalogs have search tools. These could be combined to create a better and enriched search. The EURD platform provides a pseudonymization tool, which helps not only to pseudonymizing patients at the local level of the registries, but provides also the tools for data linkage, meaning identifying if one and the same patient is registered in different registries, in different data sources, and making possible to exchange the information about the patient from these different registries. And the same pseudonymization tool, SPIDER, provides the service of data transfer, data exchange between or directly between the participating registries. Another point of collaboration and of alignment is related to the standards for data collection. A few years ago, the EURD platform released the set of common data elements for rare disease registration. And it is worth exploring the integration with the EMA data quality framework in the context of the common data model. Next slide, please. With this slide, I would like just to show that the EURD platform and one of its core components, which is called ERDRI, short ERDRI, meaning the European Rare Disease Registry Infrastructure, works since the beginning very closely with the ERNs. There are already almost, or a big majority of the ERNs is represented already in the EURD platform. You can see here the ERNs participating, 21. And this figure doesn't relate only to the ERN central registries, because at the level of the ERNs, there are two types of registries. There are the central registries of the ERN, and there are the contributing registries, the local registries, which build the ERN. So the platform is open and encourages all the registries to join it. Not only the central ERN registries, but also the many hundreds of local registries. And with this, I will stop and thank you for your attention.
Simona, thank you very much. My sense is huge potential from these resources. And as you've emphasized, the EMA catalogs, which will be launched in a couple of days, actually, enhanced catalogs which use metadata, so fingerprinting, to identify and characterize different data sources. That link will be very important. And just to say in front of this audience today that we will be sending out information to you all on those catalogs and encouraging you to register registries. Yeah, you can say that. Input data about your registries in that catalog if you wish the registries to be known, findable, etc. The last of our introductory remarks comes from Christina Kyriakopoulou, who is a senior scientific leader at DG Research, and is joining us online. Christina, are you there? We have some beautiful slides on the screen, which would strongly suggest you are.
Good morning. Sorry.
Hi, Christina, we can hear you, I think.
Good morning.
Good morning. How is Brussels? How is
It's,
Brussels?
It's sunny. So it's very comfortable here. Very nice to be with you. And I hope I will try to telegraphically mention some of our experiences at the EU research and innovation programs about the use and the value of multinational registries in translational health research, bridging as a tool to bridge basic knowledge to accelerate clinical development. And do you see my slide?
We do indeed. So go ahead, Christina. Looking forward to it.
Here I would like to say that traditionally at the European Union, we have been supporting real-world data and registries for a long time, since going back in 2000 back, starting for the using of the registries for long-term longitudinal follow-up of patients in order to be able to understand better the disease pathophysiology and the
Christina, as Peter here, there's a few people who are following online who wonder whether you could go into slideshow mode just to make it easier to read. Perfect.
No, thank you very much.
Back to you.
So here I would like to say that during the different framework programs of research and innovation, I would like to refer to the recent one, Horizon Europe, about some actions that we have been supporting in the space of real-world data, and particularly in the use of registries or electronic health records and or synthetic data in regulatory decision-making and health technology assessment. We have heard earlier from our seminal speaker, Professor Moll, about the reference to the Project MORE. Here, we have established a new real-world evidence cluster in order to support the regulatory decision-making process and health technology and or health technology assessment. There is a cluster of 6 projects. Actually, a new one is co-funded at the end of this year in order to be able to provide and develop evidentiary standards of how to better use real-world data or synthetic data as a result of the analysis with artificial intelligence methodologies in order to complement, for example, clinical trials or, for example, how to use real-world data for augmenting randomized clinical trials or presenting synthetic data or complementing single-arm clinical trials. These are some examples among others in this cluster of projects. And in the next slide, if I go here, how do we see registries in the different developments of the different European multinational projects and the development of patient or clinical registries as a source of real-world data and as a source of producing real-world evidence? One characteristic of back in 2000, if I go back in 2000, the first registries that were set up at the collaborative space at the European Union, and actually I'm glad to say that one of the examples that was qualified in the list of registries, it was the one on cystic fibrosis, and that was already started and supported back in 2000 to create the Pan-European Registry on cystic fibrosis, because at that time, there was no evidence about what are the disease mechanisms, what are the difference of the phenotypes, what are the correlations between genotype and phenotype, what is the prevalence and incidence of some of the disease, what is the natural history studies, how the severity or progression of the disease is expressed in different patient groups or different groups of diseases or specific diseases. So we have set up we have supported a lot of registries, a lot of in the observational clinical trial studies, but also cohort studies, in order to be able to create a richness of data to better understand the deep mechanism of diseases, in order to have more robust, effective translational research, which at the end will lead to more robust clinical development and more effective medicines or health innovations to the society and to the patients. So what we have seen also in these registries funded at the European Union programs is that they act as a multi-stakeholder forum. They act as a bridge to bring different clinical scientists together with basic science in order to accelerate translational research. They act as a multi-stakeholder network, bringing the patient voice more emphasized into the research questions, into the disease discovery, into the patient-reported outcome measures, which become into a more trendy term recently. And now, of course, all this ecosystem development leads to better predictors, and I want to emphasize that these multinational registries, they delivered very frequently important biomarkers for prognosis, for treatment response in the real world setting, in the healthcare system, because they are recruited from the healthcare system, several of these patient samples, and they are connected with biobanks. So there have been a lot of richness biobank related activity to these studies. They are longitudinal, they are dynamic. So this means they are evolving, or you can enrich them with additional data. During the last, from 2000 and 2010, with the genomic revolution, a lot of these registries have been populated with molecular data, genomics, multiomics. So, and they delivered biomarkers, several of them prognostic of prognostic of disease severity and disease evolution. And I say that, and this the trend during the last years also is to use artificial intelligence and progression disease models of diseases. This is a new trend in some of the projects that we see, using all the richness of high quality data, patient registries and cohorts. And of course, these are very important discoveries on the deep understanding of the patient population in order to be able to assess better predictors of safety and efficacy, better clinical endpoints, and with the use of data analytics and the power of modeling to have some predictive value to be able to use, create the real world evidence that may be useful for the regulators. And here I want to say that we see a trend, probably you have heard about the European reference networks, that we see a trend of registries being a holistic ecosystem of bridging basic and translational research. And this means they can be used for different purposes. They are dynamic, they follow the patients over time, so they can be integrating new digital technologies, mobile apps, sensors, in order to bring more quantitative patient outcomes. And they have a great value to bridge all the pathway on the life cycle of the product development or a health innovation. And I think I want to say also that to praise the data quality framework that was published recently. It is a fantastic result of the Big Data Steering Group, and several of these European registries, they follow and they build metadata models, standards of models that they could really follow this guidance that was published by EMA, and to be able to progress and to bring it to the next level of usefulness. And thank you very much.
Thank you very much, Christina. So, colleagues, we've heard opening remarks from different leaders around the European regulatory and research environment. It's my great pleasure to hand over to Patricia McGettigan to make the link to session 1.
Thank you very much, Peter. So before we start session 1, something I think that we've heard, the word that resonates with me from what Christina's just said is bridging. There are a lot of bridges that are in the process of being built. The bridges between the European organisations that organise registries, if you like, that catalogue them and so on. But we've got several of these, but we can see how we can bridge across them. And we've started that, but today is a big opportunity to carry it on. The other bridging that happens is exactly what Christina's mentioned at the end, the data quality framework, the registries qualification. There's so much overlap between elements of these that I think we can also leverage and capitalize on the benefits going forward, because I think all our eyes are open now as to the benefits of these and where we can go, especially in the context of things like the value and the capacity to utilize big data and to use to our benefit artificial intelligence for querying some of the qualitative data that are out there. So that brings us then to session 1, where a good place to start is always from where we are. So, starting from where we are, it's my great pleasure to ask Peter and Francesca to jointly take us through to the next session, Status Quo on Registries for Regulatory Purposes. Thank you very much.
Thank you very much, Patricia, and good morning, everybody. My name is Francesca Day, and I'm the head of the Therapeutic Area Department here at the EMA. And I've got the pleasure this morning of co-chairing this session with Peter Mol from the MEB here in the Netherlands also. I also have the pleasure of introducing our first speaker, who actually needs no introduction, because I understand he was introduced yesterday, and I don't think you've changed overnight, no? So, so I hand over the floor immediately to Bruno Sepodish. Over to you, Bruno.
Thank you very much, Fran, and good morning, everyone. It is a pleasure to be here now in the quality of a presenter. So if I could have my slides, please. And today, my intention is to share with you in these 10 to 15 minutes that I have. The title is, what are the regulatory needs in terms of registry data? But perhaps we could just have called it, what are the regulatory needs? And most of all, what I will share with you today is a bit of what regulators, as part of a survey we did, regulators feel about this kind of data. And it could be an interesting basis for discussion. Next slide. So again, just to set the scene from where we came, and even from yesterday, as we know from our side as regulators, and I'm now speaking more in my role as CHMP member, RCTs remain our gold standard for providing information regarding drug efficacy and safety. But also, when we look at Marguerite paintings and the way Marguerite reminded us that whenever we look at an image, we don't have, it could be a misleading representation of the world. The same way we can create an analogy with randomized clinical trials that, although very valuable and the gold standard, they do not represent the totality of reality we are faced with when a product is on the market. So therefore, real-world clinical practice is better reflected if randomized clinical trials are complemented with further data. But so these days, what we are, and the example of today and yesterday, is that the value of real-world data is increasingly acknowledged, and this has the potential to transform, accelerate, de-risk our decision-making, both pre-marketing authorization and post-marketing authorization settings, improve efficiency in the design and conduct of trials, and of course, increase our public health impact, especially when we talk about innovative access to innovative medicines. So in terms of marketing authorization, we are used to deal with this kind of data to contextualize study results, to ensure generalizability of results to the target population. And in a post-authorization setting, of course, we've been appreciating real-world value mostly due to the ability to refine the long-term benefit-risk balance. Next slide. So again, and I stole this slide from Peter Arlett sitting here next to me, and this was shared with us as the vision for clinical evidence in 2030 in a brilliant presentation. And I take the opportunity to highlight in bold 2 important messages from this slide. First of all, we hope and we aim for having clinical trials that will remain core and vital for approval or decision making, if you want to make it larger, but they will be bigger, they will be better, they will be faster. And also, real-world evidence should be enabled, and the value should be established. And this is the road we are now going through until 2030. And again, many other aspects in the slides are relevant, but for today, keep this in mind. And this is where we as regulators are currently standing. Next slide. So of course, this one is quite obvious, so given the audience that we have in the room, I will not go through them, but we know the differences, we acknowledge the differences, I was going to use the word fight, but it's not a fight. Our attempt these days is to actually make sure that everyone understands that one is complementary to the other and should be seen as such. Next slide. So real-world data comes from various sources. Today we are here discussing disease registries, but they actually, as I just mentioned, are seen by us as a complement to the evidence provided by randomized clinical trials. Also, we have seen that regulators have largely relied on patient registries, and we've traditionally used these kind of data for safety evaluation, long-term benefit risk, but we are being exposed to real-world data across all phases of drug development. And if this real-world data becomes evidence or not, that's our work. Next slide. So again, and this is also, I stole this from Peter, sorry, Peter, but this is also a nice overview. And when we look at all these acronyms of committees that we all know, I think all of us, including the herbals, all of us are being exposed to this kind of data. So the way we use it, the way we deal with it, needs to be openly discussed, and that is why these kind of workshops are extremely important. And from a CHMP perspective and SAW perspective, and of course PRAC, represented here by Sabine, this is extremely important. Next slide. So the main areas for which real-world data analyzed can support EMA committees' decision-making can be summarized in these aspects that have been already presented through yesterday and today. Understand the clinical context, support the planning and validity of studies, and investigate associations and impact. Next slide. So when we think of unleashing the potential of registries for decision making, we have to see the possibilities of pre-marketing authorization use and post-marketing authorization use. So if we focus first on the post-marketing authorization use of registries, indeed we've been dealing with this for gaining better knowledge of the medicines or the treatment's characteristics, refining the benefit either quantitatively or comparatively, qualitatively, increase safety data, further optimizing treatment, the case of optimizing posology, or even identifying surrogates for treatment effect. When we move into the pre-marketing authorization setting, mostly we've been using registries to understand disease course, the natural disease course, or the disease course with the standard of care, and the identification of biomarkers useful for the studies that lead to the decision making. Next slide. Several examples exist. We selected a few, Zolgensma, where data from a single-arm study was the base of the approval, and the data from 2 natural history studies provided information of the natural course of the disease. For instance, this was for spinal muscular atrophy that we will speak more in the next panel. Rybelsus, semaglutide, for treatment of adults with insufficiently controlled type 2 diabetes as an adjunct to diet and exercise. Here, the pharmacovigilance activity post-authorization considered also the use of real-world evidence. And finally, more recently, Nulibri, where the outcome results in patients treated with Nulibri were compared with historical data from studies involving patients who did not receive Nulibri or any other treatment. So examples are there, they're publicly available, and again, this is proof that we are continuously exposed to this. Next slide. And now I bring you a bit more a connection with More Europa project. I'm sitting next to the project lead from Groningen University, Professor Moll, and this was already introduced a while ago when we mentioned when the European Commission mentioned the MetReal initiative, where all these consortia get together to enable to establish value and enable the use of real-world data with a specific focus on decision making, either useful for regulators dealing with marketing authorization and changes post-authorization, but also downstream regulators as HTAs. We had a kickoff meeting last year, and today I will share with you some preliminary results of a survey conducted by Work Package 1, and of course with that we are also using it in Work Package 5. And this web survey wanted to get insights on the views of different players on the role of real-world data and patient registries in their line of work. Next slide. So this is a courtesy of Sita de Vries, who's sitting here with us, and the More Europa Working Package 5 team, who kindly prepared these slides. And we had the responses for 108 regulators, and we excluded from these 108 for now HTAs, payers, industry, and academia, who are considered others. And this separation is important for the discussion we will have ahead. We have a nice representation of regulators between 40 and 60 years old. Work experience is very balanced, but more than half actually has more than 10 years of experience, more than half of these responders. And then we have other responders that include HTA, industry, and academia. With a nice geographic dispersion over Europe. Next slide. So when asked their agreement with the definition of patient registries, a large majority of these regulators, 85%, agreed with the definition of registries. Next slide. When asked about the agreement with the definition of registry-based studies, again, a large majority, practically 89%, agreed with the definition that we are currently working with in terms of registry-based studies. And next slide, and here is where it gets spicy. In your view, when asked, in your view, how informative are patient registry data for studying the following aspects? And we can see all these aspects on your left-hand side that are, well, these are the buzzwords we've been hearing yesterday and today, but in the end, it's quite interesting to see that the large majority of the regulators agree that patient registry data can be rather informative to study the disease epidemiology, long-term effects. Third place, you get safety. But then when you move down, you see a tendency towards neutral in terms of comparative effectiveness. When we compare this with the others that are not the core regulators in terms of the decision-making at pre-marketing authorization, post-marketing authorization, next slide, you will see these blue dots, and you will notice that, interestingly, these other stakeholders, if we look at long-term effects, they really have, there's a difference. They really believe that patient registry data can be very useful, or rather informative, closer to very informative in terms of long-term effects. And we look down to the comparative effectiveness, they are more open to this. They believe that this information is rather informative, or closer to rather informative. Next slide. So when asked about did you view each of these aspects as a potential weakness or strength of using data from existing patient registries for decision making, again, when looking just into regulators, this has seen as a slight strength is the scalability for the relevant patient population, the costs, but then slight weakness appears to be data quality. And this is fundamental, of course. Next slide. When we compare this to the other regulators, quality is more neutral. So data quality, the perception of data quality for these other regulators appear to be more neutral. There's also some punctual differences in the aspects of knowledge, data access. But when we look to the other aspects such as scalability and cost, they are rather equivalent. Next slide. And then last slide with data, when asked in terms of familiarity with guidance documents, the large majority of responders from regulators are familiar with EMA registry guidelines, with the EMA data quality framework, with the NCEP guidance, and many others below. But interestingly, when we asked about the use of this guidance in their line of work, when assessing registry-based evidence, only 52% admits using the EMA registry guideline, and 37% mentioned none of the others. So, or none of the above, meaning all, which is interesting. It's an interesting reflection. Next slide. So, towards the end, in summary, real-world evidence supporting the evaluation of medicines across different phases of development has been evolving, driven by the ability to answer specific research questions. There is large agreement with EMA definitions for both patient registries and registry-based studies. In general, patient registries are considered informative to study disease epidemiology, long-term effects and safety, and some other responders are a bit more positive regarding its use for assessing these aspects of long-term effects and comparative effectiveness. Scalability for the relevant patient population and costs are considered slight strengths. The weaknesses relate to for patient registries appear to be more related to data quality and data access, and generally other responders are more positive for these aspects, except for the costs. And finally, regulators are familiar with EMA and NCEP registry guidance, but there is less actual use. Surprisingly, large numbers, as I just showed you, reports using none of these guidances. Next slide. Finally, take-home messages from my side. Real-world evidence has been used and will continue to be used. We cannot afford not to use this data. This was something that our chair of CHMP mentioned in a communication a few months ago, and he's right. We cannot avoid this, and we don't want to avoid it. Data or registry quality and transparency is crucial, and our discussions always end up around the quality. It's a powerful tool when used in complement to RCTs. In chess, RCTs would be the queen, the gold standard, but remember that any chess has many moves, and sometimes the pawn can be transformed into the queen, meaning that other sources of evidence can become also as important as the queen. This is a chess game in the end. Enabling the use of real-world evidence and establishing its value supports safer and more effective use of innovative medicines, and it will remain our aim. I hope I was able to set the scene to give some food for thought and reflections for the next panel. Thank you very much.
Thank you very much, Bruno, for kicking us off. And I hand over now to Peter to introduce this second topic.
So we're moving on to the second part of the session. And we will start with...
Very hold on time.
Yeah. So very good. So this fits with what. We will continue with talking about a couple of more case-oriented work now, and I'm really pleased to introduce Kieran Breen as a first speaker. Kieran and I have worked a long time already also on the patient registry initiative. He's currently also on our advanced therapies committee, but is also a head of research, also blending that regulatory and academic work. Kieran, I think the floor is yours.
Okay, thank you, Peter. So I'm going to briefly introduce this basically case study that was initiated by the Committee for Advanced Therapies, or the CAT, looking at the use of registries to study spinal muscular atrophy. So if we go to the next slide. So basically there are 3 medicinal products available on the market at the moment for the treatment of SMA, and these are disease-modifying therapies. So we have Zolgensma, Spinraza, and Evrysdi. And these came on the market. The first one was in 2017, which was Spinraza, and the most recent one in 2021. So this is obviously, it's a very rapidly evolving scene. And as the diagnosis and treatments are changing rapidly, we really need to better understand the natural history and progression of the disease, its standards, its diagnosis, clinical care, and the impact of the disease modifying therapies. So it was agreed that one way of doing this is to make better use of patient registries to see how the disease modifying therapies is actually having an impact on patients, on the disease, and how the modifying therapies is actually having an impact on patients. So if we go to the next slide, please. So in the 2022 CAT work plan, looking at how we can best make use of real-world data in decision-making, we decided to look at patients' course of disease and standards, and look at the standard care of delivery on an ATMP relevant disease entity, and we decided to focus on SMA because obviously it was a rapidly evolving area, and we had these therapies which had been approved, 2 ATMPs and 1 non-ATMP. As I said, it was an area which had developed over a very short period of time, from 2017 to 2021. There was a relatively limited amount of data available on medicines availability, its long-term safety and effectiveness. We needed to capture real-life data on patients to understand the delivery of health care and also its impact on the course of disease. And we felt that the study would inform the CAT assessment of future ATMPs, both for SMA and how the impact it was going to have, and also evidence generation, but also the potential for future therapies and the future disease-modifying therapies for advanced therapies, gene therapy, cell therapy, etc. So if we go to the next slide, there were a number of objectives of this study. So the first was to describe existing patient registries to collect data on SMA in the EEA, so to get an overview of the registries, to get a detailed overview of the characteristics and how well they interrelated. So obviously there were a number of registries out there which were collecting data, both patient registries and clinician-based registries. So how well these were interacting together. We knew that there were collaborations, there were groups, but how well they could work together to get the data and could provide this in some sort of meaningful format. The second objective was to understand how this could collaborate within a registry-based study, so to understand the registry's overall experience in collaborating with third parties, including non-profit organizations, academia, industry, CRO, and regulators, to describe the administrative processes for collaboration, to describe the operational aspects, and also the governance aspects. And we'll hear that when we get to the next speaker, that there were challenges with at least one registry trying to actually access the data from one particular registry. If we go to the next slide, the third issue was to access registries suitable to take part in the study, and the feasibility, including suitability of identified registries, mapping the data elements, again, if there are a number of different registries which are taking part, how well do they interact with each other? What is the issue? What are the potential issues with interoperability, possible overlaps, possible gaps, and how well we can make sure that we can maximize or optimize the extraction of data so that we can answer the questions that we're trying to answer, basically. And finally, to develop the study protocol to analyze the SMA natural history, its clinical management, and ultimately the impact that the disease-modifying therapies are having on SMA. Because essentially what we wanted to ask was, prior to 2017, we wanted to look at the untreated condition. Then we had the introduction initially of Spinraza and then the other two therapies in 2017. And what impact were these disease-modifying therapies having on the natural progression of SME over this time period? So to understand how the patients are diagnosed, whether this has changed over time, what was the progression or the course of progression of the disease at baseline, through its course, and then how had this changed once the disease-modifying therapies had come into place. So if we go to the next slide. So essentially, this was we agreed then finally on the ultimate aim to describe SMA characteristics at baseline, throughout the course of disease, the clinical management and its evolution, and we wanted this to be in at least 5 European countries. The secondary objective is how can we work with the registries to improve evidence generation and ultimately to look at the secondary use of retrospective data collected in SMA registries. So if we go to the next slide, these were just the procedural steps. It was an EMA-funded study, so we went through EMA procurement rules, strict timelines, etc. It wasn't a formal regulatory procedure, so it didn't have to go through a formal endorsement of deliverables through the Committee for Advanced Therapies. But as it was, as the study was sponsored by the Committee for Advanced Therapies, there was a CAT subgroup involved throughout the procedure. In February 2022, we put out the procurement procedure protocol. 2 tenders were received, and in April 2022, the contract was signed with the research organization Aetion in collaboration with Treat NMD. Go to the next slide. This was submitted by Aetion to EMA on the study protocol in November 22. The initial aim was proposed by the organization was a registry-based cohort of SMA disease to describe the natural history of SMA, the evolution of SMA care management, considering new disease-modifying therapies, GMTs, and assess the safety and effectiveness of these GMTs. We discussed this and we came up with a slight revision, and that is a registry-based cohort study of SMA disease to describe natural history of SMA, the evolution of SMA care management, and disease progression, considering the new disease-modifying therapies. And in addition, as a secondary objective, Aetion was also requested to highlight in the final study report the study challenges and the lessons learned from performing the study within these registries. So this, I hope, sets the scene for why we did the study and the remit that was given to the registry holders to carry out the study. And now I'll hand back to Peter to introduce the registry holders who actually did the study.
Thanks. So thanks, Kieran, for that work. And we could indeed, if we go to the next slide, I'm really pleased to introduce Neil Bennett from Treat NMD, who's going to present also on behalf of, I think I pronounce it, Sung Lee and Nicolas Deltour on really the practical aspect of this. Neil has really wide experience working with registries in the field of neuromuscular disease and is currently the global manager of the registry. So Neil, please.
Thank you very much. Next slide, please. So I'm going to present the details of the study you've just heard an overview of, and this is a presentation both on behalf of Treat NMD and Aetion. Sadly, Aetion weren't able to join us today. If we take the next slide. So just to kind of formalize the kind of study design and objectives a little bit, the research question we were trying to tackle in this study was to investigate the course of disease in SMA patients, but also to look at the evolution of care over time, with a particular kind of focus on the effectiveness and safety of those disease-modifying therapies. And we wanted to do that in multiple European countries using data routinely collected in existing SMA registries. Within the study, there were obviously some objectives. The first was to assess the registries working in this area, learn what lessons we could. More specifically, though, we wanted to look at the natural history of SMA, describe that by SMA types, across a broad variety of patient characteristics, motor function, respiratory, skeletal deformities, all things that are relevant to SMA. We also wanted to describe by SMA type the evolution of the diagnosis and treatment methods. And so the study design was non-interventional retrospective cohort study of SMA patients, and that used 6 registries within the federated Treat and MD network. Next slide, please. So just by way of introduction, the Treat and MD network is a global network of registries active across the neuromuscular space. In SMA, there are 37 registries in the network collecting data in 34 countries. For the purpose of this study, we limited inclusion to the European countries, and that gave us 17 registries in the countries listed here. Outside the Treat and MD network, 2 countries, France and Italy, were also contacted. They weren't able to take part, so that left us with 17 registries that came forwards into the study. Next slide, please. So this was the first study of its type that the Treat and MD network had done, and obviously to meet the requirements of the study and learning about this kind of study in a registry network, it was really important to do a detailed feasibility assessment. And so this slide just shows a kind of workflow, if you like, of that feasibility exercise. It took part in several stages. The first stage was to work with leading clinicians in the area to define key variables that are really needed to monitor SMA patients. That might be SMA type, it might be age, but also the medications and disease outcomes. We sent a pre-feasibility survey to all 17 registries that we'd identified. This was a relatively high-level summary, if you like, mainly assessing whether a registry would be able and interested to take part in the study. That left 10 registries who then moved forward into the kind of detailed feasibility assessment. And this was, as I said, much more detailed. It examined the processes in place at the registries, data cleaning, audit, data governance, as well as looking at the data itself. So as a federated network, we don't have central access to the data in registries at Treat and MD. The data is held by registries. And so this kind of feasibility assessment is really important because it gives us the kind of opportunity and the ability to say whether the data held is actually fit for purpose. And this method is called the SPIFTY method. It was published back in 2022. Next slide, please. And the outcome of the feasibility methods is shown on the slide here. Effectively, it's a heat map. You can see the 10 registries across the top. And down the side here, there's different variables grouped by type. And you can see registries in dark green are collecting those variables well, and some registries don't collect some of the variables, and those are shown in gray. If we take the next slide. And so as a result of that exercise, we went from the kind of 37 registries in the network to the 17 in Europe. down to 7 registries that passed the feasibility assessment, those that held data that was fit for purpose for this study. Kieran mentioned that one of those registries was unable to take part, and we'll come on to talk about that in a little while. But sadly, the Swiss registry couldn't take part in the study. And next slide, please. So this slide just gives a little bit of an overview of each of those registries that was selected. There were 6 registries, both clinician and patient-reported registries, collecting data in 9 countries. So some of the registries collect data across more than one country, specifically the Czech and Slovakia registry, Germany and Austria, and UK and Ireland. You can see that the registries have been around for varying periods of time. I think the UK and Ireland and Germany and Austria registry are the oldest. And you can also see that the number of patients there from each registry. And the way the study was set up is reliant on the kind of treat and MD infrastructure. So each registry is independent, controls its own data. When we deliver a project like this, we ask registries to send their data into our central data warehouse. And in this case, all registries were able to do that. The German and Austrian registry provided their data in aggregated form, just because their data protection or consent are slightly different. But the other registries sent data into the central data warehouse in a harmonized way so that analysis could take place centrally. Next slide, please. This slide shows the distribution of patients that were taking part in the study. There's just over 2,000 patients in total, 2,188. And you can see that 780 approximately patients came from the clinician-reported registries and just over 1,400 from the patient-reported registries. In terms of the treated and never treated groups, there were 1,321 patients that had been treated with at least one disease-modifying therapy. 847 had never been treated. And in some of the registries, the proportion of patients who were never treated was higher than that of the treated. You can see the German registry contributed most patients. And if you look down towards the bottom, you'll see the 1,341 treated patients compared to, 1,341 compared to 1,321. We had to remove 20 patients from the analysis because we didn't have a start date for their treatment. You can also see the different number of patients living with different types of SMA. Approximately 40% of patients with type 2 SMA, 35% with type 3 SMA, and approximately 20% with type 1. And I think probably the bottom point on this slide is probably important to highlight. So overall, 29.7% of patients were lost to follow-up. In the never treated group, that was 55.9%. And in this study, loss to follow-up was defined as 2 years without data in the absence of death being reported. Next slide, please. So if we start thinking about the objectives I talked through earlier, if we start thinking about the natural history and disease progression of SMA, by SMA type, in type 1 SMA, we can see that 101 patients achieved sitter status, 33 achieved walker status. Excuse me. The PROs we can see were 64.5 in the Spain Pro Future Mobility, Independence Pro, and the best score for active limb. It's worth saying that not all the registries collected PROs. Only the Spanish and Belgian registry were able to contribute data there. In SMA type 2 patients, 329 patients achieved sitter status, 133 achieved walker status, only 5.9 remained as non-sitters. Our median event-free survival in type 2 SMA was 143 months, And again, you can see the results of the PROs. And again, in type 3 SMA, 418 patients achieve Walker status, only 1.5 remain non-sitter status. And these statuses are, really important in SMA. They're key ways to define the disease. Next slide, please. If we start to look at objective 2, that's the evolution of healthcare over time and the evolution of kind of DMT treatment over time. So there's three DMTs approved for treatment in SMA, and you can see those in the arrows at the bottom of this slide. So Spinraza was approved in 2017, Zolgensma in 2020, and Evrysdi in 2021. And we do see an evolution of medical treatment over time. You can see that patients taking at least one DMT increased steadily at 2.2% in 2014 to 2016. Oh, thank you. And interestingly, that's before the drugs were approved. So that's presumably patients taking part in clinical trials. And we see the number of patients treated reaching as high as 68.4% in 2021. As I mentioned before, overall 1,321 patients received at least one DMT. 1,003 of those patients were treated with Spinraza, so that's 75.9%. 403 with Evrysdi and 101 with Zolgensma. Next slide, please. So now if we look at the individual drug by SMA type, in the top left panel here, you can see patients treated with Spinraza over time. Again, different types of SMA patients. In the top right panel, you can see Zolgensma treatment, and in the bottom left, Evrysdi treatment. And so Spinraza was the most frequently used therapy. Spinraza and Zolgensma usage was often reported amongst patients with SMA type 1. Higher percentage of patients with SMA type 2 were treated with Evrysdi. And the number of patients treated with more than one DMT was low in this group of patients, starting from 34 patients in 2020, increasing to 69 in 2022. Next slide, please. So to conclude the results, the results of this study were consistent with existing studies that have evaluated the progression of SMA disease. We saw clinically relevant gains in motor function in SMA 1, 2, and 3 treated patients. And the study exemplified the use of registries in rare diseases and really showed that they can provide complementary information and new avenues to answer regulatory questions. That's not to say there weren't some limitations within the study and that we didn't learn lots of really important and useful lessons. If we take the next slide. So if we start talking through some of the limitations of the study, as I talked through, we did a really in-depth feasibility study with a pre-feasibility phase, and that was really to drill down a little bit into the quality and the completeness of registry data. However, once we analysed the data, obviously we don't have access to it on an ongoing basis. But during the analysis phase, we realised that actually the feasibility phase that we'd done wasn't probably detailed enough in some ways, in that we observed that the missing data can actually have an impact on the analysis depending on which variable it's missing from. And so in never treated patients, it might suggest that a less regular and accurate, in never treated patients, we found that there was probably a less regular and accurate follow-up and probably an underreport of data in such patients. And as I already mentioned, a significant number of those patients were also lost to follow-up. We found that adverse events and deaths were underreported in these registries. It's important to say that this study took place through the same period of time as the COVID-19 pandemic. Obviously, ventilation and death are key outcomes in SMA, and so it's important to highlight that. And it's important to say that pooling pooling data in these registries, although there is a common data model, pooling the data may have introduced some imbalances and different weighting into the statistics. But we think overall the study was very helpful. Next slide, please. In terms of what lessons we can learn, but also how we can improve in the future. So the EMA registry-based study guidelines and the SPIFTY approach we used really helped in guiding and instructing the assessment of feasibility and on the suitability of these registries for use in a regulatory context. At the feasibility stage, further quantitative counting and higher granularity in defining required data could be considered. That would really let those the links between data quality and specific variables be better accounted for. The study timelines, the contribution for registries could be improved. Early dialogue with registries beyond a specific study, moving to a kind of engagement approach, I suppose, rather than a transactional approach. And also engaging more earlier with other registries. So, for example, in this study, France and Italy were unable to take part. The registries could be more involved in developing the study concept and protocol. This is something we're starting to work out within the network. We're trying to share those documents with registries to ask for feedback, to make sure the protocols we're developing would work. I mentioned that serious adverse event and death was probably underreported in the registries. And this probably ties in nicely with the next point on linkage. So obviously there needs to be better efforts to collect this data. It makes it difficult to use this data for safety purposes without those things. But we also know that those items are often collected in other systems relatively well. And so there might be an opportunity to link registry data with other data sources to provide a more holistic overview of patients. Next slide, please. In terms of the logistical challenges, I mentioned that Switzerland was unable to take part. And I think looking back, I think we've learned key lessons there around on how early we should engage with registries about studies, but also about the questions we should ask registries. So the feasibility exercise in this study asked registries, for example, about timelines for ethical approval to take part in the study, but didn't ask when those timelines started from. And there's really, looking back, probably obvious things that we can learn, but having clearly set out timelines with each registry, engaging the registries regularly is really important. I think the complexity of describing cohorts shouldn't be underestimated. Even in a network with a common data dictionary, common data model, it can be really, really difficult to describe once you're starting to stratify cohorts. That was a real challenge and something we're still working to understand. And I think there's definitely a case that maybe a common dictionary would be quite useful in not just our network of registries, but much wider. I just wanted to go back to the slide, the top point on this slide about data quality and assurance processes. I've mentioned a couple of times that we found that missingness in data was really important. And so as a network, we've already started to look at how we can better investigate this, monitor this. And if we go to the next slide, I'd just like to end with a little slideshot of a tool we've developed after this project, but also in kind of the ongoing qualification advice process that we're going through. And this is a data quality tool that we developed for registries. They can add their data to it. It'll tell them whether it's missing, complete, whether the data is coherent, reliable. And so I think there's lots of things we can do from a technological point of view to address some of these questions that we've identified. And so with that slide, I'll finish talking. Thank you very much.
Thanks for that, Neil. I think that was really impressive and a lot of information. I'm pretty sure we will have a good discussion on that in a minute. I think we first continue with the presentation. So it's my pleasure to introduce Mencia de Lemus. She's hidden a bit in the corner there in front. Camera will pick her up, but she is the previous president of the SMA Europe, but also very experienced with how we as regulators work as a representative in the CAT. So Mencia, please.
Thank you. Good morning. I'm going to be presenting now some of the lessons learned with our pilot study. I will be building on some of the things already said by Neil. I have picked the lessons that are more likely to be applicable to other studies and other diseases. I will present them in a more generic way, but I will tie them to the experience gathered from our exercise. So next slide, please. One of the first lessons that I would like to share with you is that although we had taken all the steps by the book with this study, and we had made all the efforts to tick in all the boxes, still the study faced significant challenges. So we did go through important delays. It took us much longer than expected. We encountered difficulties when analyzing the data, as Neil has just explained. And I think that an important lesson learned from this is that however clear and neat guidelines and training are provided to registries, direct engagement between the regulator and the registry holder will be necessary. And the sooner this is done, the better that will be. The complications that we faced came by the fact that each registry was limited by its nature, by its objectives, or by the laws and registrations that might limit or delay its capacity to engage with the regulator. So new authorizations were needed by ethics committees, like the case of the Swiss registry that was finally unable to participate because of the delays needed. The collection of some data that was defined as very important for the study, such as adverse events, would need new authorization of the national regulator, like in the case of the Spanish registry. Even new informed consents might be required to collect some of the data. So it's not just like going back and enlarging your data set, but it can get a lot more complicated than that. So next slide, please. A second learning that we had is that the planning phase can take a lot of time, and it's important that it takes the time it needs and that it is evolving. And in this planning phase, a good exchange with both patient representatives and experts, in addition to the registry holders, is important so that we can accommodate the expectations of the regulator and we can adequately shape the framework of the study so that we ensure it meets its goals. So if the scanning phase and the assessment of the registries is done in an appropriate way, the objectives will be established with good management of expectations, and the terms of reference will be designed in accordance with the existing environment, and we'll look to optimize it. Again, in this phase, patient representatives and expert clinicians can provide valuable information on the research questions or the idoneity of the approach. Like, for instance, through the input of patients, we replaced studying the best motor function at the moment of the genetic diagnosis by the best motor function at the beginning of symptoms, because it was explained the variability of the time to diagnosis might not might give that data not that usefulness. So another example could be that we shouldn't look at the wheelchair use as an indicator of deterioration, because some subgroup of patients were in such an acute state that could not bear a wheelchair because of respiratory issues, and therefore being able to use a wheelchair is actually a sign of improvement. Another thing is that different registries may collect different important information. So some of the registries that were used in this studies collected data from before the introduction of DMTs. Some of them collected patient-relevant outcomes. or collected, or some of them collected the same datum, but in different ways. So we found an issue, for instance, with different definitions of walkers. So a harmonized data model proved to be very, very useful. Just another lesson learned on this chapter is that the registry study, we found it to have a really iterative nature. As the data was being analyzed, the research questions were revisited, as Kieran has explained, and new ways to analyze data came forward. So it is important to have this flexible approach when conducting the study, because if you can accommodate these adaptations, the outcomes will be much better. Next slide. The third lesson learned is also an important one, is about long-term relationship with registries. So in SMA, DMTs only exist since 2017. And as registries were designed before treatments existed, it takes your time before they can start capturing data that relates to this new situation. So in SMA, it was difficult to predict how the disease would evolve once therapies started to be applied. There were changes in the phenotypes, like early development of scoliosis of patients that would not have survived without DMTs. Even the classification of SMA in subtypes, which is in the basis almost of every study on SMA, is starting to be no longer applicable with newborn screening or early treatments. Also, an impact on care by receiving DMTs, patients are more likely to attend their hospital visits. All of these registries were not prepared to capture it. So the important lesson is that the way the regulator relates to registries should not be similar to the process of drug assessment. So instead of looking into a registry, assessing whether it meets or not our regulatory needs and making a decision, if we would establish a relation with registries that lasts in time, given that all the obligations of collecting data for a disease that is receiving new DMTs, I mean, it takes a long time. It would be a much more rewarding experience. So with a good mutual understanding, the possibilities that a registry captures the information needed for regulatory decisions will increase significantly. Therefore, we should nurture our relation with registries that are capturing quality data and explore the ways in which the registries can evolve to meet our needs. Next slide. Another lesson learned is how we could have made use of the patient-relevant outcome measures that were contained in 2 of the registries that participated in this study. And also to give the importance that self-reported registries have as well. When a patient-reported registry has sufficient quality, the information it contains is comparable in quality to that of a clinician-reported registry. When we get to patient-relevant outcomes, we learned the lesson that if we want to make a good use of it, the way that they're going to be analyzed and used must be specifically incorporated into the study protocol, because PROMs can provide very interesting and complementary information to the clinical data, but they have a very different nature, so they must be looked upon in a different way. And next slide, please. Final lesson learned, registries need support. So many registries do lack education and training on the regulatory needs. To support the regulatory needs with the data that you already have, you require effort and resources. And if you have to adapt your registry to fulfill the regulatory needs, you will require even more effort and more resources. Therefore, accompaniment, training, and resourcing of registries should be carefully considered. And then finally, registry holders should be reassured on the correct use of their data. To build up a registry and to fill it up for many years with good quality data takes an enormous effort, and therefore registry holders should be reassured that their data will be used in a wise way, and they should always have the capacity to input and to correct the analysis. And next slide, I think that's all. Yeah, so thank you very much.
So thank you, Mancia. And I think that takes us to the last speaker in this group of the case study. And that is Simon Bennett from Biogen, really going to talk a little bit about his perspective on how he interacted with the regulators and the key issues that came up during the discussion. So, Simon.
Thank you. I'm very pleased to be here as part of this very important workshop. I won't spend too long going through the slides, but just to clarify, the perspective that I'm giving today is the single company's perspective on working with the registries in SMA. We weren't involved in the study that's just been presented by my colleagues, just to be clear about sort of the differentiation there. So if you can move to the next slide, please. So as everyone's heard, we have now 3 products authorised in SMA in Europe. So the first product was authorised in 2017. That was Spinraza, which was developed by Biogen, and that was partly the reason why we've been tasked with sort of presenting the industry perspective on working with the SMA registries. And just to take a step back, obviously the clinical trials that we conducted with Spinraza for the registration of the product, they were conducted in a relatively homogeneous patient population, which was obviously highly selected to demonstrate the benefit-risk profile in that group of patients. But at the time of the authorization, it was acknowledged that there would be value in collecting additional data and analysing further data on the disease to see how it progresses, given the very progressive nature and chronic nature of the condition. As you've heard, there has been a number of SMA registries were already in place at the time of the authorization of the first product for spinal muscular atrophy. So the intention was really to work with existing registries, expanding those registries to make sure that we had a disease registry approach which would be able to be expanded to incorporate multiple partners and collect high quality real world data, really to meet the needs of the key stakeholders which are listed there, and also to gather robust information on clinical outcomes, characterize the natural course of SMA and the evolving phenotypes, and improve our understanding of approved and emerging treatments in the real world setting. Next slide, please. Okay, so some of the key considerations that were made at the outset of establishing the global collaborative network of SMA registries are listed on this slide. So the intention was really to establish collaborations with multiple existing SMA registries to utilise their observational data. We also sought to strengthen the capabilities of existing external registries to fulfil the research needs of multiple stakeholders, as I previously mentioned. And it also involved building consensus across different organisations to collect new data fields as needed. And since 2017, when the registry work was first started, it's continued to evolve and involve talent from the industry and also from the registry partners and the regulators themselves. And over that time, we've been able to work with the registry holders and registry partners to improve the capacity and capability of registries to collect reliable patient-level data. We've also worked to standardise data across the clinician-entered registries to an aligned international aligned core data set. So there was work undertaken there on development of data dictionaries with TreatNMD. And we've also, as a company and other companies, have provided financial support to implement the data collection and ensure sustainability of the registries. And over time, the network of registries has evolved, and we've had to accommodate or been able to accommodate national registries which address payer needs, and some new registries, particularly this example in Germany, were initiated over the course of the last few years. And the aligned data set that we're working with the Biogen supported registries collects key information on patient demographics, clinical characteristics, medical history, functional outcomes, hospitalizations, and treatments. Next slide, please. So this slide just provides a snapshot of the coverage of the registries in April 2023, so it's fairly up to date. So We have 9,500 patients across 25 registries in 28 countries in the Biogen-supported registries. And this obviously has grown considerably since 2017, and we're continuing to add patients to the registry as we move forward. Next slide, please. And the value of the registries in terms of decision-making and supporting regulatory decision-making is really that it provides insights into the natural history of SMA as the disease continues to evolve with the treatments that are now approved, and also the effectiveness and safety of those treatments over a longer period of time. These data were presented as a poster, and I can certainly send the link around for the poster, but just a couple of areas that I wanted to highlight just because of the context of the discussion today. You can see that from the registry's work that we've undertaken that approximately 40% of the enrolled patients were adults. And this reflects the discussion that we've been having around the additional information you can get and the additional value from registries, because typically adults are underrepresented in SMA clinical trials. The majority of the patients that are enrolled are type 1, type 2 or type 3, and that's fairly evenly distributed across the 3 types. And also these findings have been used to support reimbursement submissions, but they've also been used to support regulatory decisions. So the European label for Spinraza was updated in 2022, and the SMPC section 5.1 now includes information on the treatment of adult patients with type 2, type 3 SMA, and some of that, the data package that went towards supporting that regulatory decision included information from the registries. And I have to say that's despite the registry that we're working with not being qualified. I know that was a point that came up yesterday. If you move to the next slide, please. Okay, so as we reflect on our experience to date and the sort of learnings that we can carry forward, it's apparent and very clear from our work and our understanding that not all registries are equal. The data quality and consistency varies between different registries, even within the same disease area. And given this and the fact that registries need to be fit for purpose or fit for use to address specific questions, it may be that some are able to generate data that can answer questions for the regulators and other stakeholders, such as an expanded patient population in SMA, whereas others may be more appropriate for more general trends around epidemiology, treatment uptake and natural history of the disease. But going back to the qualification question, irrespective of whether the disease registry has been qualified or not, registries aren't a shortcut to regulatory decision-making or decision-making by any stakeholders. They involve a significant amount of work, and we just wanted to make sure that this was sort of, I know registry holders and a lot of people in the room understand that, but it's when people talk about real-world evidence and registries, it's considerable effort from all parties involved, and it needs to be maintained over time. So there's a list there of sort of activities as a medicine developer that we've undertaken. We now work with the registry, so evaluating the data collection process, evaluating data quality, overseeing registry expansion, initiating and reviewing contracts, and discussing with other stakeholders and companies. And in some cases, the actual data quality that you receive versus the expected data quality can only be ascertained once the analysis has been performed. So that again involves looking at that data, what it's delivering, and then going back and working with the registries and with the regulators to see what needs to be changed and updated. So this has impacts beyond the marketing authorization holder, obviously. Move to the next slide, please. Okay, just briefly in conclusion. So I think from our experience as a company working with the SMA disease registry, it's clear that disease registries have demonstrated opportunities that exist to address key questions. They also are able to accumulate additional scientific knowledge and broaden the understanding of the disease. However, that really depends on the availability and the quality of the registry data that we can obtain and that can be produced. And any steps to support this are fully supported. So some of the tools that we've started to talk about in this workshop around the data quality framework, the feasibility assessments and the checklists, I think are all steps forward that can help to improve the data quality of registries and ultimately deliver registries that can answer the questions that regulators are asking companies to address when the products are on the market. The other piece that that I just wanted to highlight again, and goes back to one of the points that Mencia was mentioning, is around early engagement with registries. So I think Biogen's involvement in the SMA disease registry started in 2017 once a product was approved for the treatment of SMA. I think there's potentially areas where we can, and I mean this as a sort of as a community, we can identify disease areas where perhaps no products have yet been approved, but we can perhaps look forward and see where they might benefit from early engagement between registry holders, regulators, and medicine developers for early collaboration to make sure those steps and processes are put in place. Because certainly some of the learnings that Mencia mentioned around some of the challenges and some of the long timelines of getting registries up and running are certainly reflected in our experience from the industry in SMA. And I'll just conclude there. Thank you.
So thanks. I see one hand raised already. I think, well, maybe just start with that and then I'll give the floor to Francesca to kick off the real discussion. So please, I can't see your name, but.
My name is Christine Dehn from German Heart Foundation. I have a question regarding what you just said, Simon. The information presented, you said that the drug was researched in clinical trials for children and or minors. So in the real world data, you aggregated data for adults. Is that correct? So, and how did you present this in the package leaflet information? Did you mention explicitly this has been aggregated by real world data, or how do you present that information? Or did I misunderstood anything?
No, you understood correctly. The clinical trials for the initial authorization included pediatric patients. The label change in 2022 included information for healthcare providers in 5.1 around the use of Spinraza in adult patients, and the package leaflet was updated to reflect that fact in 2022. But we didn't make specific reference to real world data in the package leaflet.
Okay, thank you very much. And thank you very much to all the speakers. I think it's there's nothing like a use case to really bring concepts to life and really see what the challenges are in real practice. So thank you very much for that. And thank you for bringing already a lot of lessons learned, because I think that's key also for the ongoing discussion. So we're probably going to eat into your coffee break a little bit, but we'll try not to be too long with that. But we now have some time for some questions and answers, and I already see hands up. Just to mention that people online should be able to put their hand up and ask questions in the chat. Is that correct? Yes.
In the chat.
In the chat. So please, if you're online, ask questions in the chat, and in the room, of course, you're very welcome to lift your hand. So let's start.
Thank you very much. Good morning. This is Viviana Giannuzzi from Benzi Foundation, and I'm also a PDCO member representing patients. I would like to ask the speakers some questions on their experience so far. And the first one is for Bruno. That considering your experience within CHMP and COMP, how much a registry that has been qualified by the EMA really makes an orphan drug designation or a marketing authorization application smoother and quick, considering the qualification. And the second one is considering the experience in the SMA. One of the main issue is addressing consent challenging, because of course we need to have a valid granular consent to use the data for research purposes, to process data, personal data in compliance with the GDPR, to possibly share data outside the European Union. We heard that this morning that some non-European countries have been involved in collecting data, and to possibly share data with the for-profit organization. And so patient possibly should consent about that. Thank you.
Thank you. If I may start, thank you for your question. Yesterday, actually, at our breakout session, we had one specific session dealing with that issue, and I think that there might be others here who might want to jump in. I think we all agree that the qualification process brings a needed discussion with the registry holders in terms of what we would like to see to character for that context of use. And we had this discussion with some of the colleagues here in the room at the time, is that the qualification is given to a determined to a certain context of use to answer a specific question. If that later translates into a smoother movement at these committees and the comp works, perhaps when we mention the comp is not such that the designation phase, it is useful, of course, if we have data on the clinical practice, but it will be extremely important later on when we want to maintain. It's usually the maintenance phase of the orphan designation at the time of marketing authorization. And there we have a few success cases. We presented here today a few orphan drugs and just mentioned those. But even for them, and I think the CAT, when I mentioned CHMP, it's implied that we are also, of course, considering the issues with the CAT. It is valuable having a qualification. I think it is. It's yesterday someone said it's a stamp that gives the registry a stamp of quality in the end to answer that specific question. So the perception I got from yesterday, from those involved in the room, for those in the room and those involved in qualification, is that from the sides of the registry holders, it's useful. From our end, I can tell you that it seems to be useful, but we even heard yesterday from Sabine that in some cases not having the qualification has not precluded the use of the registry. So we might have some work ahead in trying to find room for discussion for the registry as a whole, and then specific discussions leading to qualifications when we are addressing specific questions. So that's perhaps one of the outcomes of yesterday. So very, very generally, I think in the end, use is there, value is established, is a form of scientific advice, but in this case is a seal of guarantee. And for us, CHMP, we and other decision-making committees, we see it very positively. I hope I have answered your question. And the other one, I think, can be probably for my colleagues here.
Yeah, so just to talk to your point about consent a little bit, so you're absolutely right. So different registries, and I talk about our network here, but different registries have different patient consents. And I think when you're designing the study, you kind of have to do it both ways. So you've got to design the study to take into account what consent the registries have, but also the other way. So we're doing studies at the moment where some of the registries are reconsenting patients for specific use cases. For example, in the study that we presented, the German and Austrian data was actually analyzed in Germany and only aggregate data shared, and that was because they don't have consent to share patient level data outside of their institution. And so I think you have to be flexible with the study design to make sure that you're working within the consents that people have provided, but also sometimes that, in a big network, there may just simply be some registries that can't take part in a specific study for that reason. I think that's how we would approach it.
Okay, thank you. And I see we have some questions online, so Peter will attempt to paraphrase or read them out.
Yes, so there are quite a number of questions coming in. Some are very detailed, and I'll try to. Two questions really focus around the quality, and maybe I'll start with Sonia Zbaki's comment that asks, what is sufficient quality in practice, maximal number of missing data allowed to generate valid evidence, and a very interesting one also on the variations in geographical setting that may have impacted in her mind the analysis of subgroups. I think there was an interesting observation seeing the prevalence very different, or a number of countries being having many more patients, in the registries, then also I would believe compared to your population size you would expect. So maybe you can start on that, Neil.
Yeah, so I think the other thing you have to factor into that is the length of time the registries have been in operation. Obviously, that was quite variable, so you'd expect some differences there. The point about what data quality is good enough, I mean, that's hard. That's something we're working on right now. The tool I showed is all about monitoring what it is. The other bit you need to know is what you'd like it to be and what you're kind of working towards, and that's something we're kind of starting to do at the moment. But that's a difficult question.
Kieran, you want to? No. Okay. So okay, in the room I see Cecile. Cecile.
Thank you. So it's a question for Neil and Simon. So the work that has been done in SMA with the description of the disease, I see also synergies with other type of tools that are currently gaining a lot of traction, like disease model, which are to date developed based on data coming from clinical trials, but more and more now combined with real world data in order to have the use of the model applicable beyond marketing authorization, but also for payers and HTA bodies. And I wonder, I think that when the methodologies applied to disease models such as pharmacometrics and statistics can help to really decrease some of the uncertainties that might come from having models only generated based on real world data. So I wonder if this is something you have considered in SMA, or because maybe in some area you have disease model that exist and maybe expanded to SMA and have some tools that can be generated in a synergetic way. Thank you.
Thanks. So, I mean, from our perspective, and just speaking from Biogen's perspective, I think that's, I think it's a really interesting point that you raise, and it's not something that we can really, I can't really comment on at the moment, but I think it's something that potentially would be valuable to look at moving forward with the data that we have from the registry at the moment.
So one of the, I guess, strengths that we have is that we work across a broad number of neuromuscular diseases, and that lets us apply learnings from one into the other, and that's something that this study has been really useful in that we're now applying the learnings into DMD and other diseases even further down the pipeline. In terms of the specifics around data collection and how you can kind of drive that from one to the other, that's tricky. But I think what we're starting to see is that there are probably, how do I describe this? There are probably similarities that we see between, say, DMD and SMA in terms of the shortage, no, not the shortages, but fields that are less well collected than others, for example. So I think the lessons we're seeing here are probably applicable in those diseases. My guess is they might be much more widely applicable, but I can't say that with any kind of evidence or certainty.
Thank you.
So we're going to alternate a bit of the questions from the room and the online ones. And one is a nice kind of challenging comment where I think the whole panel could respond to is the question is, should a patient registry always be kept in an academic organization? So maybe we'll start with Bruno.
I do not dare. No, I think but that question is also connected to another one that I saw a while ago with the funding. So for me, one goes very well with the other. So depends on a lot on will the academic have the ability to keep it. So that's something for consideration still.
I'm not sure. It's not. I don't think it's a question of where it should be kept. I think it's a question of access. And I think that's the key thing. From I guess from a regulatory perspective, a patient perspective, and an academic perspective, it's really a question of and even an industry perspective, it's really a question of access. And I know of registries who have been funded by industry, but at arm's length, and which can be accessed by purely by academia, by even by patient organizations if necessary. So I think it's really just a question of access and openness and transparency, and I think that's the key aspect from my perspective.
I agree with Kieran, and I would add to the access bit the part of having a good representation on the management board of the different interests. So an academic registry can perfectly accommodate a patient perspective, ensuring that their clinical data that are capturing is relevant to patients. So it's captured in a specific way that is pointing out the important aspects of the disease for the patients and for their life. So yeah, no, there are no barriers here. It's just how we collaborate all together.
So I just add one more, as well as access, I think you need to consider where the data is being sourced from. Certainly within our network, there's academic kept registries who are patient-reported registries. So I think that's the other distinction. Kept is a slightly odd.
Okay, thank you. And I think Francois in the room was first.
Thank you, François Ordis. Well, thank you for these interesting presentations on this SMA registry study. In clinical trials, when a clinical trial center is excluded from the analysis, it is carefully explained. And I wonder if the same rule should apply to registry studies. There are some registries who cannot participate, for example, because their data don't fit with the study. But for those who don't want to participate, I think this is something that we need to analyze and understand how we can change this to make sure that we have all the data we want and we need in these studies. And then I have two questions. One is how the results were explained to the registry participants, or what are the plans to share the results of such analysis to registry participants. I think it's vital to inform the registry participants, those from the 8 who took part, but also the others, that such studies are being conducted and what we learn from these studies. In a way, it's more important than the consent for secondary use for the patients. And my third question, it's in line with what you asked, Bruno, I think. Was there an economic analysis to assess the overall cost, all costs included, to perform such a study? Thanks.
Yes, I can talk a little bit about the sharing of the results. So the reports, well, the results themselves and the reports coming from that. Let me start again. The results and the report generated from those results have both been shared back to the registries. Those registries can then share that with the patients who are part of their registries, and part of the work we do at Treating MD is try and support those registries. If they need materials and things, we'll work with the registries to answer that.
I'm feeling brave. Maybe just taking 3 steps back, Francois, I strongly support your methodological point. And I think it's not just specific to registries, it's specific to observational research in general. So as we know, there is a formal process for approval of clinical trials, and no clinical trial can be of a medicinal product can be done in the European Union without going through a clinical trial approval process. That's not the case for observational research. However, having a pre-agreed protocol and analysis plan would seem to be extremely important in terms of driving up quality, but also preventing phishing exercises and so on. So it's a general point, not specific to registries, but I think in observational research, we do need to work collectively to drive up the methodological quality, if you like.
I think Christina's had her hand up for a long time, so we'll go to her.
Thank you. I have a different question or comment. This issue of informed consent is of critical importance. And of course, these lessons learned from this case where different consents, in some case, precluded the inclusion of some data or some registries, might be an eye-opener for the future, meaning many of these consents were not drafted with a specific intent of not sharing the data. They were drafted just because somebody that wrote them thought it was the best text they could find out. So it's not that was by design, they were created like this. It was a bit more like happenstance. And so I would think that it would be a great service that a template for, and maybe already existed and I'm ignorant, but a template for informed consents of registries should be put forward. And of course, people might be willing not to follow it, but at least they would have where to start, because this is a huge issue. It's an important topic, and sometimes it blocks completely the use of the data, completely. And it was not the intent. In fact, most of the patients, large surveys among patients are all say they are willing to participate in research and they are willing to contribute to research. So in a way, the consent that was written by somebody that was not a patient makes the data not usable. So my suggestion is that in the, let's say, bucket of interoperability, I know that's more a technical issue, but the consent is not forgotten.
So, Christina, I think really important point, and I'm going to look in a minute around how people respond. It's one of the things that we discussed also when we designed the More Europa project. Really, we have an ethics group on board from the Charité, trying to also work with them around how you can consent, especially in Germany. There's obviously a lot of discussion. They have worked a bit on a sort of kind of general template, and it is a thing that we would also like to probe a little further how we get there. But I'm looking at my colleagues, if anyone want to give this a step as well. But it's clearly that we need to work on a sort of toolbox around how to facilitate this. So there's another question in the chat, which is recurrent, and it was already asked a bit before. I didn't want to skip it, but it's a complex one as well, which is about the collection of the adverse events, especially those adverse events of special interest. If they are going to be added at some stage in the data set, we will get perhaps missing this is some of the comments. And further down the discussion, which I think is a true point. A question again is on if you add information or if you add data collection to something that is not routine practice, that becomes something that is sometimes falling under the trial regulation. I was wondering how, if anybody could reflect maybe on how registries try to stimulate that this is being collected just under normal practice.
So I think lots of registries have very close relationships with both the patients who participate and the clinicians who submit data. And I think there's an engagement piece there as well, where those registries can work with those stakeholders to try and not so much drive, but maybe support those people in reporting that data. I talked about linkage for those things that maybe are challenging or not collected routinely. But I think there is a certain amount you can do with just by supporting, but then we add in support at the top level where we'll support registries with bursaries from our governance committee and things like that, where those bursaries might be to support the registries to do that piece of support with their stakeholders. So I think there's a certain amount you can do there. Yeah.
Okay, go to the room. I think I see a hand there. Or you have to put your hand up high because I can't see very far. Okay, over to you.
I do have a question for Bruno, and you said yourself that some of the results of the survey that you presented were spicy, so I'm going to take it from there. You showed that there's a high awareness of the various guidelines for registry-based studies, and also like various guidelines that are closely associated but this is not routinely used or so highly used by the regulators. And the question is, did you delve a bit more into the why behind this? Thank you.
Thank you very much. It is a very good question, but I also told you all that these were preliminary early results. We are actually diving into this, and this is part of the analysis the working packages are doing. And this is actually something we got pretty much last week, but these are quite interesting. And exactly those questions, they need to be answered. And again, I saw one question from Xavier in the chat, which was also connected, for instance, on how we define the guideline, meaning when people reported no use, do they mean that they don't know? But one thing is whenever we make this question, we assume that even if people are working and if they know the EMA guideline, they might not go to the guideline every day, but they use it in their head. This is what shaped their line of thinking. So when people reported not using, I think we need to explore what do they mean by not using these guidelines, and specifically how can we further address this. But this said, I think we need to really dive into more of this spiciness. But these are interesting results, and this is why we wanted to share with you today. And I think the other point that I think we also need to share is this difference between we separated HTAs because they were in a reduced number of responders compared to the others, but and we aggregated them with the universities, so academics and other stakeholders and industry. But it's good to understand that there is more openness or more positiveness to the aspects of this of the real world data that we still do not see in the regulators. So we need to work in that direction. So again, for this specific audience here today, I'm not preaching to the converted, but when I go back into my world and with Peter and in my other world, I think this is definitely a strong message that we need to reflect on and understand how can we move this positiveness towards a more closer image of what we are seeing with other regulators that are more open to this kind of data. So I don't know if you want to compliment Peter, but
I think you were quite good on that. I think Peter actually had a point.
Well, you said you didn't want to preach, but my grandfather was a preacher, so I'll take them. I think we need to go back to basics a little bit. I'm talking about the guideline, uptake of the guideline. I think we're often quite good at writing guidelines as regulators, but not necessarily very good at change management. And I think there's a bit of a mea culpa moment here, because I think it's a beautiful guideline, the registry-based studies guideline, but we didn't roll out training tools to help support people to use, etc. And I think one of the things that I would like to come out of this workshop is not to say that we need maybe we do need new guidance, but to build on what we've already got and to further implement it. Because I think it was all wrapped up in COVID-19 pandemics, we've got lots of excuses, but I think that guideline needs to be implemented and we need to train and engage. Thank you.
So maybe to wrap that up, because I think really it's a good point that Peter is making here. And I see also a comment of my colleague, well, in the meantime, it's my friend, Paolo, the last one, but Marcel Qua, who is a pharmacovigilance assessor at the Dutch agency, and really says there's a lot of observational methodology already out there, and that's obviously being applied also by many of the regulators. The question is specifically, if we talk about registries, when does that fit and when does it come into our decision making? It comes at various different aspects. We have about 4,000 regulators in Europe working on very diverse diseases. Obviously, for every orphan disease is half a regulator or something like that. But you can imagine if we get in a dossier suddenly, or not suddenly, if we get these kind of data, they are different from the traditional trial data. So we have to work towards also implementation. I think that's what's one of those aspects of the Horizon call, where this material cluster was working, is also really heavily working on. It was really, there's so much work ongoing in your field of real world data that also the translation of how to apply and look at these data best is obviously something making use of various tools, integrating in the work process that indeed Bruno and I are sitting at, Francesca also daily, where it has to be integrated in that work. So I think that's really something that I take up. A lot of issues you've mentioned we haven't touched on. There's a lot of things going on in the chat that is, I think, very good. We'll try to maybe at least copy it and take it along for further thoughts. We will not be able to answer it at this stage. I'm not going to talk longer. We are going to go for coffee. I really want to thank the panel for their talks and for you for your attention.
Okay, welcome back after this short coffee break. So I'm Patrice Verpilla, I'm the head of the Real World Evidence Workstream within the Data Analytics and Method Task Force at the agency, and with my co-chair, Sabine Strauss, who is chair of the PRAC, a member of the Dutch Regulatory Medicine Agencies. We will chair this session number 2, which is on experience gained and hopefully lesson learned from initiatives aiming to leverage the use of the registry. So we've seen already this morning with Treat and MD how this could be the case, but you will see that how we can implement also data quality into this experience, and then we'll have a concrete example with Harmony at the end of this session. But let's start first with Kit Ross. Kit is a professor of biostatistics at Radboud University Medical Center in Nijmegen in the Netherlands. He is chair of the Methodology Working Party at the European Medicine Agency and senior assessor of the Dutch Medicine Evaluation Board. So please, Kit, the floor is yours.
Yeah, thanks Patrice, also for the kind introduction. And maybe I can have this next slide, please. Yeah, my topic for today is fitness for purpose and the data quality framework. I will, next slide please. We'll try to take you through some highlights of the data quality framework and the thinking behind it. Although I think also from the earlier presentations, some basics are already quite well known. But I particularly also will reflect on how it fits indeed for purpose for regulatory decision making and explore, at least that's what I will try to do in the time allotted, to what extent it can help in the discussions we also had yesterday in terms of qualifications of registries and easing the road to using data from registries for regulatory decision making. And a few takeaways, which are personal points of view, most of them are actually, but also maybe to steer here and there some discussion on our qualification process as well. Next slide, please. Across yesterday and already today, there has been reflection on where registries can be useful for regulatory decision making, and the typical areas of augmenting the evidence from clinical trials range from supporting planning and validation of applicant studies, understanding the clinical context, and investigate associations and impacts. We've seen examples. I noticed in the discussions yesterday. We did not necessarily in all our discussions took this broad perspective. There was a relatively large focus on the well-defined post-authorization efficacy and safety studies, because they provide a very clear context of use for registry data, but the use is much broader. And I think it's actually essential to consider the broader perspective also if you think in terms of data quality for registries. Next slide, please. And I'm going to admit guilty here as well. I'm part of the More Europa project. And in this case, I actually want to use it to illustrate the richness of use of registries. And it's also good to realize within the MedReal cluster of projects, this is the one that actually focuses on the use of registries for regulatory decision making. So it's not an entire coincidence that we come back to this project a couple of times. This is actually to help position the registries in a proper way for scientifically valid decision making in a regulatory context. And I will take you through some of the things that we will actually be doing to illustrate the diversity of research questions a registry could be used for. And I start with early development. One of the things that we will be exploring is to what extent can you use data from registries, and we're using the big MS data registry as an example, the Swedish heart failure registry, as well as the oncology registry that Peter mentioned yesterday from the Netherlands, as examples, to see whether, let's say, biomarker modeling in association with clinical outcome based on registries can actually inform early development decision making. Because typically early phase clinical trials are done in biomarker short-term outcomes, potentially surrogate outcomes, and within most companies, some level of predicting probability of success of the full development is informed by those trials. And you can actually use registry data, we think, to improve that decision making. If you look at the phase before decision making, it's, as mentioned, contextualization, of course, of the disease. Direct augmentation of clinical trial data with parts of registry data, actually strengthening the evidence directly for clinical trials. Typically we say external control, but to be honest, I don't necessarily like that term. I always want to know what's the actual study design in total that aims to answer the research question. And external control doesn't really tell me how you want to use that and what your research question is and what your proposed research design is. So that's one area. And one step that we usually hint to, that always happens, we always extrapolate what we've seen in clinical trials in phase 3 to a target population. And we're always aware that that target population will differ from the population we've seen in clinical trials. That's not per se a problem. Because we are able to extrapolate. What we do very rarely is actually make that translation truly quantitative. Use modeling and the clinical trial data to really predict what will happen in a target population, which is slightly different, has older people there, more vulnerable people. And some information on how that would behave is there in clinical trials, but we don't do that very quantitatively and very scientifically, if you ask me. So that's those areas are we exploring. And if you look beyond approval, some other uses we will make there is target trial emulation for observational studies. For me, that's one of the, let's say, methods and approaches that can sincerely help very much improving the methodological quality of observational studies that Peter was alluding to. Registry-based clinical trials and modeling for HTA support. Pretty much along the same lines we would do for generalization for the regulatory decision maker for approval. Similar modeling should actually and can be done based on registry data for supporting reimbursement decisions. And as we've also seen across the 2 days, it's usually not one registry. I think the most valuable ones will actually be collections of registries where countries and national borders play a role. So one of the other things that we will be exploring is federated influence. Can we leave the registries where they are and essentially have smart algorithms pass the registries, making analysis across multiple registries a lot easier. So that's to give you a flavor on how we think registries can be used. And I think the next slide is probably the most important point I would like to make for my thinking. If you think in terms of quality as fit for purpose, all these uses may impose very, very different quality requirements. And I think that was part of the key discussion we had yesterday as well. Christina chose to have a very confined, clear context of use to define the quality requirements, but that means it's not qualified for everything else. And I think one of our my let's say takeaways here, and I will get back to that in a bit more detail if we go to the framework, is can we disentangle some core quality and qualify that and allow a registry to be qualified for answering more than one question? That will be hugely helpful. But it doesn't necessarily fit our current thinking. But I think this is, as a start, what I wanted to illustrate in terms of what can registry data be used for. The next slide, please. And I think actually the data quality framework can truly help to some extent. I'm guessing most of you are at least with some basics aware. I think on the left-hand side, there are 2 important ingredients, at least in my view, in the data quality framework as is. And those are the determinants on the left-hand side. And there is a clear distinction between, let's say, foundational parts of quality. And that's the way the data is actually collected and how it matches the original source and the original intention of that data. Intrinsic qualities, which could relate to the level of how things are in statistical terms. How are they formatted? How have you made choices in terms of the granularity of reporting age, or how do you report comorbidities? Fairly intrinsic quality, and question-specific parts. So it's a totally different question whether you want to see an association with a biomarker or whether you want to augment the clinical trial for phase 3 late-stage decision-making on core clinical outcomes. That will be very question-specific. So I think that the data quality framework clearly makes that distinction. And I think actually that should be helpful also in the way we look at the qualification of a registry. Because the foundational qualities and the intrinsic qualities might serve more than one research question and more than one context of use. So I think that's one important observation from my side. The other one are the quality dimensions, and I found it interesting to see it in the treat NMD example of the dashboard. I actually saw them, if I correctly, and those are the things that you typically associate with quality. Reliability it's actually, does it correspond to what's in the source data, or are there very peculiar data there? Coherence, are data analyzable, which is, if I look across registries, are formats similar? If I indeed look at the ability to walk, is the ability to walk indeed the same across all the registries? Am I looking at the same data? So and that's a very basic quality, irrespective of the context of use, that needs to be in order. Likewise, timeliness, particularly for safety-related questions, you do not want the registry to lag 3 months behind in terms of the data that's there on safety. So but also that's a fairly basic requirement which is valid for more than one context of use. There are two which are more closely related to the context of relevance. Is this the data that's actually capable of answering the research question? Is it relevant? And is it sufficient, the extensiveness? So there are things like missingness, and the level of missingness can become important if missingness is not entirely clear. For instance, whether if a particular event like mortality in the treat and NMD is not there, does it mean that the patient still lives, or is it simply because I don't know, which is a fundamentally different outcome? Those are the dimensions. Not all of them are directly related to the context of use. So there are some core elements there. Next slide, please. Oh, this is 2 in a row. They look alike, but they are not entirely the same. Now, from where I come from, registries serve multiple research questions, and I think most of you will face, many may actually not be known at the time of design. So I really like the point made in the previous talk. It's dynamic. It's not a constant over time, a registry. You will add stuff. You potentially will also change variables because of growing insight and harmonization across registries. What the data quality framework proposes is to actually have a tab, and that's why I did like the dashboard kind of view of the treat NMD, the SMA example. Across the different dimensions, from reliability to relevance, have a tab on metrics there, on how do you actually assess at what level you are in terms of the different dimensions of quality, and have metrics in place to assess that. Across reliability, extensiveness, coherence, timeliness. And you see that relevance, pretty empty in terms of metrics, but that's really only related to is it suitable to answer the research question? I think a fundamental point that's in this table as part of the quality framework, and I think that's where I think we always need a close discussion between regulators and the registries. I really appreciate the question, could you be clear in terms of the requirements and what is an acceptable level of quality? But I hope I've made clear that that's a hugely difficult question to answer in general. Because essentially that is driven by the research question, and that is driven by the potential regulatory impact. As I said, if it's used for early development decision-making, the regulatory impact is much smaller than when it's actually used to augment the clinical trial to reach a certain level of evidence to enable the primary approval decision. Regulatory impact is totally different, so the acceptability level on what's acceptable quality will be at a different threshold. But I think building your registry and having a clear tap on where you are in terms of data quality across these dimensions will allow, let's say, a statement on for which context of use or for which questions this does meet requirements, and that can be more than one context of use. I think that's the discussion we need to have to facilitate registries better than we are doing now, probably with qualification procedures and advice. Next slide, please. I'm switching a bit closer to the information that was also shared with you on beforehand, and that's the annex more closely, because the data quality framework is essentially for all data. There is an annex related to real-world data, which advises on how could you use parts of this data quality framework, particularly for real-world data, for quality assurance, quality reporting, and data quality assessment. And this matrix is proposed, and you see the relevance column is not there. This is really the foundational part of data quality. And the suggestion is make sure that you have indeed in place, if you look at the rows, the different parts of your quality system on your registry that tackle the reliability, effectiveness, extensiveness, coherence, and timeliness. And the idea is, if you look, it's a fairly simple matrix shape of template, but it will help you to test, to see whether you're complete, whether there are gaps, redundancy, or where you need to do something complementary compared to what you're doing now to reach the right level of quality. But these are the parts which are, and that's the thinking behind the data quality framework, not directly related to the relevance question. That's the next part of the system. Next slide, please. And this is the quality in relation to a specific research question. And here the idea is, and this is quite similar to actually in general attacking a research question. You define your higher level research question, but then you take a closer look on what's actually achievable within the data I have available within this registry. So it's kind of the general part, but also the high level relevance assessment. And in line with what Peter proposed, you operationalize your research question into an actual protocol for your study. And what's actually also quite common in clinical trials, it's not that different. Your question might actually not be directly answerable by your research, by your registry. So there is an interplay between defining your research question, looking at what your registry is actually capable of doing, and then maybe you have to be, and usually it's in one direction, become a bit more modest in terms of the question you are really able to address given the limitations that are in the registry. And this is a natural process, I think, and that does not make the question you end up with, which is answerable by the registry, it still remains very valuable. And the framework annex that's for registries also proposes using, and I saw already happening with the Treat NMD framework, do a structured fitness for purpose assessment, and also there SPVIT is proposed as an approach there. But for me, this is very close to, this is how you do science in essence. But this is on top of your foundational quality of the registry. Next slide, please. A few points to make. Some of them I've made to some extent already before. It is difficult to pre-specify minimum criteria for data acceptance because it depends on the type of study. It's disease dependent. It's also analysis dependent because some analyses are more robust than others to perform. This is kind of within the research question. On the other hand, it might be as good as it gets. So the consideration is if there's nothing or very limited data out there, that also influence your decision what will be acceptable or not, because we do have to move forward in the interest of patients. So also that's one of the reasons why it's very difficult to pre-specify acceptability criteria. You might actually hurt yourself by doing so. So conclusion on relevance, as a conclusion, it requires an in-depth systematic assessment against design elements, and the annex actually has some concrete tables, templates, etc. to do so. I think I also kind of was triggered by the remark, what if you go to subgroups or similar subsets of the full source? Actually, some of the quality, particularly in relevance quality, might be lost. You might actually not be granular enough. The fact that you have the full population there, but you would love to know something about the elderly, might give a totally different cut through the different registries you have. And you might not. So that's also where I think we need to think about how we approach qualification of registries. It's not only versus the context of use, it's also what part of the registry will you actually use for the research questions you have. And if you subset or do subgroups or make different selections, you might lose quality, in fact, particularly in terms of extensiveness and coherence. Okay, and then I'll conclude by giving my takeaways, if I recall correctly. Next slide. Oh. So given what, I think actually given the potential that's there in registries, my takeaway would be aim high for the foundational dimension of quality. Get the basics done really, really well. And I think we are still also there in terms of evidence. It's a benchmark, but also clinical trials in terms of data quality remain a benchmark. Because I think the better you are there, the more likely you are able to address multiple research questions based on the registry. I think we need some thinking about the qualification process to reflect these different levels of the different levels of determinants and the flexibility to answer multiple questions. I truly struggle, but that's for discussion, with defining a very limited context of use, because it also gets lost in translation. The idea is we qualify for a very limited context of use, but the message out there will be this registry is qualified. Which is kind of difficult to interpret. And this is not unique for registries. I've also seen examples of algorithms that are qualified where exactly the same happens, where we have to limit the context of use tremendously, but the message that gets out that this algorithm is qualified. Yeah, yes, for a very, very, very specific purpose and not in general. So I think we would help ourselves to be clearer here and open up a way of qualifying that it's clear that multiple questions are answered based on the same registry. And a last point for discussion, we had lots of discussion on yesterday in my group on review, tapping into, are we still qualified? I would kind of see that more as a continuous improvement process. In the sense, if you do systematic fitness for purpose assessment with each and every new research question or each important new research question, it's actually also an opportunity to improve the registry. And that's the kind of cycle I think we could discuss about, and that could also determine the frequency or the way we interact between registry holders and regulators. When do we want to tap into whether it's still qualified? Yeah, I would. That might also actually be driven by answering new questions rather than having it on an annual, regular basis because it's so nice to plan in time. So that's suggestion. And so the takeaways for discussion later on. Thank you.
Thank you so much, Professor Roos, for a very insightful and interesting presentation. And some of your remarks really resonated quite well with me. I have now the honor and the pleasure to introduce two speakers who will take us through data discoverability. We had already a sneak preview this morning. And the first speaker is Anna Coquino, and she's a medical doctor working in the Data Analytics Task Force at EMA, leading the activities around strengthening the role of real-world evidence in medicine regulation with a focus on data discoverability and data quality. And Professor Franz Schäfer, who is a professor of pediatrics and chief pediatric nephrology at Heidelberg University Hospital. And he's also currently the president of the International Pediatric Nephrology Association. So I think this will make for a very interesting presentation. Please, over to you.
Good morning, and thank you for this presentation. Thank you for having me this morning here. I'm here on behalf of our team that's working on the HMA EMA catalog of real-world data sources and studies. So I'm here to present to you what we have been doing for the past almost 2 years that comes now to a conclusion. Next slide, please. But starting from the beginning, though, just to put things into perspective, where we started from is the EMA HMA Joint Task Force, the Big Data Task Force and the Big Data Steering Group, who presented us with the recommendations of the direction to travel in terms of real-world data. And what we have done is to particularly focus with this project on the third recommendation, there called data discoverability. So in going towards enabled data discoverability, what we have done is first to go to identify some key metadata that is required for regulatory decision making, and that was the ground base for what we have afterwards built, which is the catalogs that I'm presenting to you today, which is one, a catalog of real-world data sources and a catalog of real-world data studies. We have done so by going and strengthening the current NCEP resource database and the current EU PASS register. So we build up on something we already had using the input of this activity. Next slide, please. So the catalog will focus specifically on two big topics, in describing the real-world data sources and describing the studies with a set of collecting metadata that will help pharmaceutical companies, researchers, and regulators to identify and use such data when investigating the use, safety, and effectiveness of medicines. So these two catalogs, as I was saying a bit earlier, they build up on the information we already have in the NCEP resource database, which means that we have migrated a number of data sources to it to start off this project. What we have also done is to bring in other data sources, and we have reached out to various data holders to bring in information on various data sources. So now we are at a point where we have a sort of a good start and to start from. In terms of the real-world data studies, we have built on top of the EU PASS register. So we have basically migrated every study that we have now on EU PASS register, and similarly enhanced the information. We went cleaned it up and so on and so forth, data management sort of activities to reach this point. It is, we like to think it's an efficient and user-friendly platform, and you will see it for yourself in a few minutes. It has a centralized and enhanced way of managing compared with the tool that was available before. Focus on transparency and observational research. It aims to go to focus on following the data principles of FAIR, to be findable, accessible, interoperable, and reusable. And behind it, you wouldn't be able to necessarily see, but behind it there is a module that is actively looking into connection with other similar projects. So it's to cater for the interoperable part of this project. We're looking into facilitating the search and evaluation of data sources and studies related to medicines, and we're looking into the future to integrate with other catalogs, EHDS, and other similar initiatives. Next slide, please. Just a few examples to understand how this would be used and what are the strengths of these catalogs. For instance, the way we built it, we have interlinked data throughout the system. So data sources, studies, institutions, they're all built into information sort of mini network in our catalog. So that means that for one, you would be able to go and simply search a list of data sources that have particular features that you're after. So, for instance, captures and medicinal product information, disease, demographics. These kind of details are all captured in the details on a data source when planning a study. What can be done is to see, for instance, the study protocol that was used when using a particular data source, or several studies that have been creating using the same data source. You would have direct access to see the protocol, the results, if they're in that stage, sort of an integrated look of the data sources and the way they are being used. The next slide, please. So from now on, I will be showing you various screenshots from the catalogs to have a more hands-on feeling of what exactly is it, the tool that we are proposing. So for one, this is the sort of landing page. You'll see it hopefully live shortly. You'll see it has a look and feel like our corporate website. It is integrated in our corporate website in the sense that we have moved it from the current location in NCEP. It is now with the rest of the content in the corporate website. And shortly after go live, we'll also have it integrated with other documents and other content that we have on our sites, such as RMP, PARS, qualification opinions, whatever else we have there. Next slide, please. This is showing you how it looks, how the catalogs look behind. So under the search and simple use that a member of the public is able to do, we have naturally a data management feature there. So this is what someone who would submit a data source would see. They would have an account, and they would be able to go through this form and fill in the information in a structured way, going through some steps, having some help, some lookups, and so on. This is largely how it looks like. You'll see that we have information both administrative, the usual, where would you find, say, a contact point, or where is the website, but also we're going deeper than that. We're going in data elements collected. So does it have what kind of vocabulary it used to capture death, or what does it have pregnancy or neonates? So there's a series of data elements that are more meaningful than simply the administrative ones. We also have a number of quantitative descriptors there. So things like how many entries, split of patients per age, and things like that. And lastly, in the data flows and management, we capture information on things like common data models, like specific vocabularies that we have for indication for various data elements. Next slide, please. Similarly, for the data studies, just to have a bit of just to have a bit of a feeling of what it would look like. That's the form on the left. So same concept. It would have a form that is controlled and with structured data elements in it. On the right, you would see the link between a data source and associated study. So you could see how you, starting from the study listed as a title, you could go to data sources and see the details of the data sources. Or you could go to the data source and see what other studies you have with that particular data source. Next slide, please. There's two data elements, and I'm more mentioning because you'll see them there. We capture also, you see, institution and networks. We capture that to help structure the data. We don't necessarily have as a purpose to have a catalog of institutions. There's several tools to track that, but what we have is a structured way of capture institution networks for this particular catalog with a minimum set of information to help better queries. So we have this institution, and for the next slide, we have the same similar with the network, so a minimum set of information for that, a description, website where you'd find, and a connection with the institutions that are part of the network. So again, a bit of a network of the way data is structured. Next slide, please. Again, just to give you a feeling of what's behind, as a user or us as validators that were looking at the data before publishing, you'd have this kind of dashboard behind where you not only are able to edit things, manage things, and so on, but you're also able to collaborate with colleagues. So we have this feature that was required especially from the studies part, so that several people are able to work on information from the same studies. Next slide, please. Just a brief look at this slide basically shows you how we would how an edit would work, and several states in which an entry could be before going to being published. For the next slide. The search functionality is indeed worth spending some time on. What we have done is to have a search that is overall for the whole website, and you're able to search in everything, but also focused on each element. So here we're looking at the search functionality, looking at the data sources. You will see for now a minimum amount of filters. So we have started from a smaller set of filters that we found particularly useful. This being the plan here being to grow gradually. The first attempts of having 50 filters didn't exactly end well from a usability perspective, and it seems like it more confuses people than it actually helps. So our approach here is to have this limited amount of filters. We have a big search term sort of field where we can that search is absolutely everywhere, PDFs everywhere. It's very efficient, and we take it from there just to sort of frame how the starting of this. And you can see here, for instance, how it looks like. So we have filtered here, say, data sources, the country to be Germany, and the data source type to be hospital inpatient records. These are the 3 results showing the filters that were applied. There's also a possibility of the button there to export results in an Excel with details of each data source that can then be filtered further. Next slide, please. This is an example of search throughout the whole catalog. You can see here that we have searched hepatitis widely, and then the results, there's 92 results. The first 2 of them is a study and a network, for instance. So that's just look at the overall search. Next slide, please. More specific and more focused for this event, we looked, for instance, we shown a search where we're going for data sources, and we select data source type, say a disease register, for instance, and we see here 59 results. Then we're going to try to do that live, hopefully. Pressing and on the right, so that's on the left, the search of the data sources and the results that come like a list of them. And then if we open one of them, we can start looking at the details. And I have particularly pointed out the data element that I hear that has been asked about a lot, whether we capture the data source qualification. We do. And this is the example where we do. I think next slide, please. So, I mean, okay, what's happening next? Well, we're going live in 2 days. That's why it's been. You will be able to browse these catalogs in 2 days yourself. We have a multi-stakeholder webinar for the launch, so we have started communicating about it, and we're going to communicate widely. So a webinar on 4th of March in which we will show in details what we have proposed as tools there. We plan to continue communication activities and raising awareness, as we know and understand that the going live is one thing, but keeping it further on is the real challenge. So we have a plan ongoing to keep focus and to keep these tools up to date and relevant. We need to finalize the integration of the information with EMA website content. It proved more difficult than we expected it to be, but we are going to do that. As I was saying, following actively the maintenance process and the collection of new data as well, we have been doing that, approaching data source holders with invites if you'd like to join our catalog. It's continuous process in terms of the fields that we collected, the lookup values and the search criteria. We had to start from somewhere. Everyone agreed that the set we have started on, it is a good one, but we are welcoming the continuation of this. We are looking on a yearly basis at least, potentially sooner in the first year or two, to keep improving this, the data set that we're collecting, the lookup values that we are offering, and the search criteria. And last but not least, we have a parallel piece of work in which we are looking into integrating the data quality work that we've been doing. So we have now a minimal sort of information on data quality, but we are looking into growing it a bit by bit. So to have more structure, to have more integrated with our other piece of work on data quality. So that's kind of the three messages, big messages that I wanted to share. That we have a go live in 2 days. That the catalog is publicly available simply by clicking. So no login necessary, as it can be sometimes a bit confusing. It's widely and publicly available, and that we have an integration between data sources and studies. I think in the next slide you'd have some links to various demos and documents that are okay. No, we can circulate those. And I think now we're at the part when I'm trying to actually demo this. So I shall move around the room trying to get access to the website. And here, yeah? No. This one. Here. That one, please. All right, I see. Okay. Not necessarily the exact, so how you would see it on your own, whatever, computers at home, as because of the WebEx, I can see that the fonts are a lot larger than usual, but it's not that necessarily that important. So this is the landing page of it. So this is an environment, of course, of testing. This is actually the first time I think we're presenting this in an external sort of setting. Come to think of it. Just a brief sort of look. Right, so we have, as I was showing earlier, we have data sources, studies, institutions, and network. In terms of the data sources, we have here a button to basically see all the data sources as a bit earlier. Wondering if the zoom of this can possibly be, I think we can zoom this by any chance to be smaller letters, because it's looks a little strange. Just trying to get the zoom because it normally you'd have more information in a page than that. Oh well. We won't just 20 seconds. If not, I'll just go through it. No, it's all right. Okay. So this is the what I was showing earlier. This is the filtering option. What we can do here is to maybe show you how the search works. So you have the values here that are available on data source type. I can search for things like registries. I can select disease registries like I did a little earlier and apply this. What we'll do is, so I can see here I have 55 results, their data sources, their disease registries, and then we can maybe look at the one we showed a little earlier in a more live and a bit details or not. We were looking at, yes, we were looking at something. So what I can do is to then search further and to say, look for Huntington, for instance, because that was my, it will have some suggestions of its own. We have 2 registries that are dealing with Huntington's disease. We were looking at, I guess, this one. And then we have various information here. For instance, it's a disease registry, it's a biobank. We have several identifiers that we have defined for this. This up here, where it says a PURI, is a persistent unique referenceable identifier that is used for interoperability. So it's the part I was saying that we're working behind to have it integrated with other sort of initiatives. It has 4 type of elements here. So it has the administrative details in terms of, say, name, acronym, who's the data holder, has it undergone a qualification, more descriptions about it, and so on. So these are the, if you'd like, more administrative sort of details when it was established. The publications also are listed here that they were for this particular data source. And then looking at other elements, perhaps it's of use. The data elements collected, I was saying that we go in quite some details. For instance, we have a coding of the disease and in the MEDRA codes. That this, for instance, can be integrated, and we plan to have it integrated with other systems and other information. So we went for structured information here. Whether the information is captured, not captured, what kind of vocabularies we use, and so on. We have perhaps the quantitative description here. It might be of use to. Okay. So, for example, we have like a split on age group or population size, active population size. So it goes to that level of details. Just to give an example. And lastly, data flows and management. And then it says governance. Where would you find more information about the governance, the description of the data collection, what triggers an entry? So that's a wide description. What triggers the registration in this registry? So when would you have a new entry? And perhaps interesting, but not entirely easy to explain, the linkage. We captured the linkage both of what data sources were used to create the data source we're looking at, but also what other data sources could potentially be used to create something else, to link to. And then it captures information, which data source and how exactly, which variable, how complete would that linkage be, and so on. So it goes into some level of details over linkage. And I think with that I should be stopping, because that's, I think, more than enough. But thank you for your attention.
Thank you, Anna. So 2 more days to go, I will say, to wait more or less. And if you're not part of the 55 registries already registered in the catalog, please do it as soon as possible. So meaning on February 15, you will be able to enter the data. So we expect to get much more in the coming days. So France now.
All right. Can I have the first slide, please?
Yes, Christina, quickly.
want to make a comment regarding the enroll linkage of data, because I think it's quite unique. It's not working as well as we would like. But the issue is we attribute a unique identifier to each participant that is called the HDID. And it's more or less like the books in libraries and in bookstores. They are the ISBN. So that's what we are trying to do with HDID. And so we recommend everybody that are doing clinical trials in HD or anybody that are doing any other study to generate the HDID for their participants. And the HDID has an algorithm that is in a server, so it can be used by anybody that is willing to do it. So once people get an HDID, and for example, they are in enroll, but they are also in a clinical trial, and they are also in some other initiative, we can connect the data from these different sources. So I think it's there are room for improvement on the methodology, but I think it's a unique approach to keep the data connected from different sponsors, let's say.
Thank you. Thank you for the comment. I think we all agree that indeed this is an important step needed for linkage, and linkage are important indeed to ensure that we have more holistic views on the data. Thanks. Franz, now floor is yours.
All right. So I have been asked to report a little bit on our experience with onboarding to the EMA catalog for real-world data sources and studies. So I'm the coordinator of ERK-NET, the European Rare Kidney Disease Reference Network, a consortium of 100 specialized units of adult and pediatric nephrologists in Europe. And we have built a registry from scratch in the last 5 years, which we call the ERK-REC, the European Rare Kidney Disease Registry. Next. So I have been told to be brief, so I put all the information on our registry on one slide. So this is a single core registry for all rare kidney diseases. It provides demographic information. It allows to identify patient cohorts for clinical research, but also to provide natural history data and deep phenotyping. And finally, also we monitor our clinical management performance and the outcomes of our patients in this registry. It's a centralized online database, and we have been able to enroll 23,000 patients in the last 5 years at 80 specialized units in 24 countries. We collect a slim data set from all patients, and then we have for some of the major rare kidney diseases, we have extensions. There's 7 of them. And you can see here the number of data items that we collect at baseline, at enrollment, and then annually during follow-up. Next. So we were approached by EMA to onboard our registry to the emerging catalog. And the entire procedure took altogether 4 or 5 months. But if you want to quantitate the amount of work associated for us with that, you would talk about a couple of days, actually. So in October 22, we were contacted by IQVIA, the CRO that was very helpful, very instrumental in the onboarding process, and we shared our metadata dictionary with them. They pre-populated the data capture sheet and made us review it. So they were able to pre-populate about 90% of the elements, and we completed the rest, which was mainly the metrics that you just saw on the example. We then had several iteration processes and cross-checked our entries, and finally we clarified the last few elements, and that was it. Next. So the main requirement for onboarding to the catalog is really to populate the data capture sheet that is shown here on the right side. It's a simple Excel form that covers the elements that are defined in the EMA list of metadata for real-world data catalogs. In our case, I don't know how that will be handled in the future. Most elements were pre-filled by IQVIA, and they based on the ERC-REC data dictionary that we provided to them. And that showed that it's very important to have a comprehensive documentation of one's own registry, because that facilitates the process a lot. As I said, we provided some additional contact information, details on the family linkage issue, and the metrics like the records by country of age, the median observation time, the fraction of active patients, and so on. I think it's also important to define the initial methodology for the metadata updates and their frequency. So we have talked about that, and we agreed on 6 monthly updates of our database or our database entry, because as you have seen, our registry is still in the linear growth phase. So we are growing by 20-25% per year. So a registry of in this phase is certainly rapidly outdated if you do not update it regularly. All right, so since I was brief, I still have a few minutes of time. So I would like to show you 2 more catalogs for rare disease registries. So can I have the next slide? That oh yes, and that was just a thank you slide and appraising the very positive experience in working with the colleagues here. Next. Simona Martin already this morning alluded to the ERDRI, the European Rare Disease Registry infrastructure that contains the European Directory of Registries for discovery, a central metadata repository for finding the metadata from all these registries, and a search broker. And virtually all the ERN registries and a total of 129 rare disease registries have currently been included in this catalog or in this system. And it would be really nice from our perspective to somehow link the 2 infrastructures in the future to make them basically cross-searchable. Next. Next slide, please. I think we can skip these. This is just a description of the directory where you can discover registries. Next. And the metadata repository where you can find which kind of information is collected by the individual registries. Next. And then the search broker where you can search for certain data fields. Next. And then I would also like to point your attention to a new development that we have been involved in as the rare disease community in the European Joint Program on Rare Diseases that was jointly funded by the Commission and the EU member states over the last 5 years. There we developed what we call a virtual platform, which is a federated ecosystem of resources that are fair by design, that aim to serve the rare disease research community, and that includes biobanks, knowledge bases, IT tools, but also registries. And we have a user interface, which we call the virtual platform portal, that allows users to query all the resources, including registries, to find those of interest to their research. Next. Next slide. And here, a little bit unlike what we have seen in the catalogs so far, you have the same function, which we call the level 1, which is resource discovery. But then we also have a level 2 that allows to discover content, to search, to query contents and receive aggregated data from the information stored in this virtual platform. So basically, if you want to know how many patients are out there in different registries with a certain diagnosis or a certain age range, you can launch a query of that kind here. And in the final stage, you will also be able, of course, with proper authentication and authorization, to perform remote data analysis on the record level. This platform is fair, as I said, by design. So that means the resources, in this case the registries, must present their metadata in a harmonized machine-readable scheme, and we use the data catalog vocabulary, the DCAT, for that. This metadata is then made visible to the outside world directly, automatically, using a system that is called FAIR data points. Okay, that was what I wanted to add. Thank you.
Thank you so much, and thanks for the nice words, having been part of the pilot with us on the real-world data catalog, and reminding also on this initiative presented this morning by Simona. And hopefully with the link catalog, one day we will have a more comprehensive view on and knowledge on the existing registries in Europe. So, last presentation by Professor Hernandez Rivas, who is a professor of the Department of Medicine at the University of Salamanca in Spain. He's also senior hematologist at the University Hospital of Salamanca and head of the research groups in molecular cytogenetics at the Biomedical Institute of Research of Salamanca and Cancer Research Center. And he is also there because he is the project coordinator of the project Harmony and Harmony Plus, and you will present us this initiative. Professor, the floor is yours.
I don't know if I'm, hello, good morning everyone, and first of all I want to thank the organizer for this excellent meeting and for inviting us to show and to share with you the results coming from the Harmony database. And also as hematologist, I really very happy to be here in front of you, trying to share with you our results in the big data platform that we have run in the last 7 years. I'm trying to share my slide, but I don't think it is. Okay, I will go with the presentation, and we will see how we will manage. Okay, next. So the basis of the Harmony Alliance was established 8 years ago was the meaning that we have a lot of data. Hematologists is a very well organized community, and hematological malignancies are not so frequent, but with a precise diagnosis, and most of the cases at molecular level, and in some cases using also data coming from omics results and genetic results. And this was the idea in which we have the challenge to unit all the silos in which the information was recorded at that time. Next one, please. Can I have the next? So the Harmony Alliance is how we call 2 EMA projects, Harmony and Harmony Plus projects, in which we have built a large community with a wide representation of all stakeholder groups. Patient organization had an important seminal role in the platform and in the alliance, and they are conducting the work package of the stakeholders. But also, this is a public-private partnership, and the industry is developing a very, very important level together with the regulatory agencies, HTAs, and also academic and hospital, and working together really to be focused, to be centered in the patient. Next one. With this approach, we have generated and coordinate a community of roughly 500 professionals working around the world now. We had over 140 data providers and 9 European patient organizations. Harmony Alliance involved 9 pharmaceutical companies. And with this, we are, so now we are collected over 100 databases and identify over 150,000 cases in patients with hematological malignancies, leukemias, chronic and acute and chronic leukemias, multiple myeloma, and so on. And we build up a robust big data platform, which is GDPR compliant. Next one. These are the numbers that in the institutions or the countries that we are participating or identify with are providing data. And this is quite strange to be in Europe, and the list is organized in alphabetical order, and it started with Argentina and Australia and finishing with states in Uruguay. But we have all the participants, or most the participants, and most the data has been collected from Germany, from UK, from Italy, and also from Spain. But I think we have a quite good balance between the North and the South, the East and the West countries in Europe. Next one. And this is in the next, okay, thank you. And here we have, which is the data that we have transferred to the platform. And you will see that there are over 55,000 acute leukemias, have acute lymphoid leukemias and acute myeloid leukemias. And we also have a good representation of chronic lymphocytic leukemia and multiple myeloma, almost 20,000 cases already transferred to the platform and ready to be analyzed in the different tools. Next one. So which are the mainly characteristics of the big data platform? So the first one, in the next slide, we will see that we have built a data lake. And in this, but now we are thinking about the possibility also to include data federated. So I think it is this hybrid model will provide the best performance for trying to get a balance between the data, the quality of the data that we can have as a secondary use of the data already transferred to the data lake, and also to have the advantages of a data federation. And this kind of balance will be the best idea, I think, to improve and to enlarge the number of cases to be analyzed. Next one. Okay, the topic of the talk was interoperability, and I think here you will see, I will say that this is more the long way for the data from the data identification and the data providing knowledge. So this is something that we learned. This is one of the lessons that we learned, that the data must be identified with enough quality. We can again come back to the quality in the let's say later on. The data that are going to the platform are anonymized. Later, we are going to do an OMOP, a common standard model, to do data harmonization. And I think there are a couple of words that in some way I missed in these 2 days, that they are data curation and data validation or result validation. And this is something that really it's a critical point. And data curation together with the harmonization, sometimes we don't have this kind of idea in the registries that we must curate the data that we are receiving, and curate 1 and 2 times or as many times as possible. In the next step, we visualize the data, and we are going to do, and the data are able to do analytics and ready to do dissemination. So it seems that it is an easy way, but if we are just having a look, next slide please, in the harmonization, we will see that, can I have the next? We'll see that the OMOP data model that is also used by Darwin and many other registries, is the way in which we can really have the same data already organized in the same level. And this is the data in which we are able to say, okay, the data are organized in the same way. But just to provide a very, very simple and schematic example, in the next slide, we will see a longitudinal scale of one of the patients that we have included in the platform. This is acute myeloid leukemia. Is a woman, 59 years old, going to receive a high-dose chemotherapy, but before we assess the rearrangement, and it was a rearrangement in an important gene, emerald gene, she received the therapy. She was in CR1, she received a consolidation therapy and later allogeneic stem cell transplantation, and she is alive almost 2 years and a half later. This is what we see, we are usually seeing the databases, but in almost the transformation of this model we will see in the next slide. And this is the numbers that we get. So the first time that I provide access to the statisticians, to different statisticians to work with the system, they reply me, okay, we are not able to work with this. In this language, it is quite difficult to do and to get any kind of advance. And this is something that we have to keep in mind, that OMOP is not a trivial tool, it's a powerful tool, it's a really very important, and this is a common language for all of us, but must be in some way refined. Next. Okay, and now I will invite you, as I have seen that most of you has a computer, please, could you go to Harmony Alliance webpage? Okay, and you will do the presentation, not me, but you will do the presentation. It is another way in which we can do that. In Harmony Alliance webpage, if you put in Google, you will get the possibility to ask and to work with the platform. And this is something that I will prefer, not for me, but for you to get access to the full platform. I hope that this is working, that you can get access. I see the screens from some of you. You can go to the green screen and you can see that in the green screen, it is 2 ways in which you can get the full access to the at least 2 diseases. So in the first 2 screens that you will get, it is AML and ALL. You can click on. Okay, so this is open. This is you can work, and you will get a scroll on the right side. You can change at any time, and you can have a look on the data, how the data are organized, how you can work with the data, how you can in some way play with the data. This will be automatic tool. It's not a Nintendo console or something like that, but this is very close, because we have also to convince the young people that you are able to do that in that way. And this is a way in which you can combine any kind of question that you will have for acute leukemia, cold lymphoid leukemia, or in the other screen, if you are going back, you can go also to the AML. It's working. I hope that this is working. Okay, so you can play. I'm not playing, so I'm not trying, okay, what are you doing? You are doing, okay? So it's a different way. So you are coming back to the web page, so you can do wherever you want. And I invite you to play with the tool, because it is really something that we develop mainly for young people, because they need to see the data, how the data are organized. If you are coming back, you will have in the third graphic, the third one is a predictor. So the third screen on the web page, we have developed based on Bayesian methodologies and some machine learning. We have based a system to really individualize the therapy of a specific patient. In that way, a patient with acute myeloid leukemia, you can identify the patient, you can see if the patient is 50 years old, is a lady with this or the other characteristics. So it is quite easy to do that. And we advise that you cannot use this because it's not a medical device. It's something that will help you to do the things and to organize your ideas. This tool is also publicly available. And I want to stress here that this tool is based on the results of 12,000 cases with acute myeloid leukemia. It's not based with 100 of cases. Now it's based with a very, very high number of patients involved in this. Okay? So this is the kind of tools that we are able to manage. It's a pity that I cannot get access from my computer to show how also we are now for the partners and for the people who is providing us research questions. With the same idea, we are able to do Kaplan-Meier curves to calculate PFS1, PFS2 in multiple myeloma patients, to add all the genetic markers in order to better clarify which are the more important commutations in a patient with an acute myeloid leukemia. But this is not publicly available. It is just for the people who is asking us for projects. So we believe that this is the way in which we have to proceed, and this is the kind of tools that I really want to share with you and also to offer to EMA in case that you are interested to work with these kind of approaches. Okay, can I have the next one? This is the analytics that you have seen in your screens. Next one is how to explain this support the decision making in this case in acute myeloid leukemia. And my last slide, can I have the last one, the next one? We are now moving to a foundation. Usually IHI projects are about 5 years. We get the funding for a period of 7.5 years. And what we are going now to do in the Harmony Alliance Foundation is to continue to offer these services to all the community, for the regulatory agencies, for the patient associations. Patient associations, for instance, lead some kind of projects inside Harmony, and they were really very much committed in the definition of the outcomes that we carried out by Delphi service in the last couple of years. So in that way, this Harmony Alliance Foundation will provide a robust, safe, consolidated, and GDPR-compliant big data platform together with the visualization tools. And these visualization tools are usually written in R and managing classical statistical tools, but also we can do this in machine learning and improving or using by artificial intelligence. I have to add here that this tool is we can customize the tool according to the needs of the different registries of the different institutions, and in that case to the EMA. Next one, please. Can I have the next? So this is the added value that we had for the different members, the different stakeholders. We really want to have the patients, and in fact, the patient organizations are co-founders of the foundation, together with the European Hematology Association and some academic institutions. And also to, let's say, the ideas or the needs for the academic or for the research side could be different from the regulatory, but the data that we manage maybe can be used in the different ways. And this is what we really want to add here and to offer to all the community, pharmaceutical companies, clinicians, patients, regulatory agencies, and HTAs. Next one, please. And now going to the end, okay, This is not an easy task. So you were managed registries. I cannot tell you all the lessons that we have learned. So it was already discussed. At the technical level, there are a lot of heterogeneity in the data. The data are sometimes collected even in paper, or in a very, very old database. Sometimes the technology is totally outdated. The relevant data sometimes are limited. I have to stress here, together with the previous 2 points, quality and validation. Another is competition, because we had a very strong competition, mostly from states, mostly from companies who are really doing business with data. What we want to do is just to improve the quality and quantity of life of patients. But our competitors are really very, very, very strong. And all the legal and political and governance troubles that I think we must agree in that way. But in any case, next one, we are trying to, together with the other hematological projects as Euroblood, NET, and many other projects in consortia working on the field, we are trying to put together all the members, all the stakeholders, because this is the only way to improve the health and to improve and to gain some kind of value in these terrible diseases who really, really are extremely, extremely bad for patients. And in the last slide, I really want to thank you for your attention. And excuse me, because I was not able to do that, but you were able to do it. And sometimes it's even better. So at least it's a different way. Thank you so much.
Thank you very much. So before taking some questions from the room, maybe just because you mentioned, Professor Hernandez, HTA bodies and payers, most specifically on the slide, just before we had 2 questions related to this on the chat. So can you envisage working more closely with HTAs, using patient registries in order to improve data and cost effectiveness? Will there be a possibility to avoid duplication of some efforts and therefore speed up processes? And the related question, it looks like the costs are not considered relevant data. In few cases, costs are collected together with clinical data. That scenario has strong implication for assessment. There is a need from HTA side to harmonize data collection efforts. So I don't know if you...
Yeah. I want to tell and to share with you that we have a couple of HTAs in the Harmony consortium. So we were 53 members. It was not so easy to manage this, but okay, in that way we were able to survive. NICE was one of the more important participants in the consortium, and now is going to become also an important member of the foundation. So we receive a lot of feedback from NICE, and I have to really thank NICE and all the Dalia and Catherine for the support that they provide us in the way to do the qualification of Harmony at the EMA. So I totally agree with this point. We have to avoid duplication, but I have also to stress some point here. The data that we obtain, we cannot tell that the data is cheap or is expensive, or which is the cost of a very good data with a good quality data, which is the real cost. So it was something that really is, so I was asking myself several times, which is the real cost? The real cost for a very important, a very good data, it will be no limit. But the question here is that we are able, and we are thinking just to reimburse or to refund a data, but also we have to keep in mind, and we have to ask, let's say, European Commission to support also initiatives to do data curation and data validation. For most of our studies and for most of our projects, we perform an internal validation because we have a lot of data, and also we perform external validation with the public data that are available. And this is the only way, because with this amount of data, to get a p-value, a significant p-value, is really very easy. But what is the real significance of this?
Thank you. So any questions in the room? There were a few more in the chat, so maybe let's go to this, mostly linked to the catalog. So which steps did you perform before the construction of the catalog? Any survey to select filter criteria? Any process? Which categories are included in the study filter? And few more to come.
No, for this one, so we had, I think, yes, a full year of preparation to get there. So indeed, we had a project with a consortium that looked at metadata used in various catalogs. They put it together. 1 year on, we had a big file of 400 data elements to choose from. We had prioritization exercises to look what is most important for a regulatory. For this prioritization, what we did is to have surveys, exactly. So we went for surveys to external stakeholders out there, we even went a bit international because more is better, sort of. So we had, I think, quite a sturdy process of getting to a list. In terms of from now onwards, we have a process in place. So we publish also the description of this process. We have a help desk. We expect requests for this catalog to grow, and then we have a process in place to manage them and to update our catalogs, likely on a yearly basis.
Thank you. Maybe an additional questions, more for both of you. In fact, can this different initiative be integrated into one platform? I know it will be a challenging one.
I think that will be challenging, but at least for the second and third platform I was referring to, we are actually working on this. We are trying to link or even merge the catalogs and the information on the metadata. And in the future, we will have to see how we can link also to the EMA catalog. Absolutely, for the rare diseases.
Can I just a brief intervention from EMA side. Having heard Simona's intervention, and there's been quite a lot of dialogue with Joint Research Centre over the years, but my sense is we need to intensify that. And there will be opportunity for synergies between the catalogs, but also more generally in terms of our efforts to support registries. So I think I'm saying publicly, making a commitment that we will intensify that.
Yeah, I think we as users would really appreciate a single entry point rather than having to enter things 3 times.
I chose my words carefully, but certainly greater collaboration to see what we can do between the different actors, because we're doing things that where there are lots of touch points, aren't we? And I think we acknowledge that.
Maybe a question for you, Kit, because if I'm well understood, you said that we should be modest and discuss what we can do when we have a research question based on the data available. Can we turn this around by saying, okay, should we just adapt the way the data are collected and the amount of data collected, what are collected, to be able to answer to the research question that we have?
I think it's both, because quite a few questions also have urgency. So you might not be able to wait to answer the bigger question that the registry is totally up to speed. But I think if you're faced with limitations in your registry data for the questions you would rather have answered, I see it as an incentive to improve. So I think you should use that research question to bring the registry up to the level that it is capable of answering. But we might not always have the luxury of waiting for that. I think that's how I see it.
Perfect. Any additional questions? Yes, Neil?
Just going back to the discussions yesterday about the value of qualification, is there any way these catalogues could really kind of highlight the registries that are qualified? So the example shown had 2 Huntington's registries. I assume only 1 is qualified, but that's kind of hidden away. Could that be like the top results or something?
I mean, for now, we're capturing the information. We could change the way things pop up. It's interesting thought. But having the data is the first step.
Your question comes 48 hours before go live, so you can imagine there's an emotional part to the response when somebody then suggests a change. But of course, there's going to be feedback, and that sounds quite important. If we put a lot of effort into optimizing the qualification process to make it more useful, it does seem like an obvious thing to do to then promote the availability of the information on what has been qualified.
And then just to add to that, and leveraging what you've just said, Neil, from the discussion we had yesterday, because there was a nice piece around that as well about highlighting in patient consents and so on around registries, that the registry you're contributing to is qualified for whatever purpose and so on. So I think we could have a sort of a unity of purpose across the things that highlight within a registry, the patient information about a registry, and the catalogues that we're talking about. So it would be a unified message there, highlighting the qualification. Thanks.
Thank you. On the back of the room.
Thank you. And yeah, just a comment. Basically, we published recently the report of the experience gate with regulator-led studies from September 2021 and 2023. In there, we report about a query from the PDCO regarding the frequency of FLT3 mutation in patients with acute myeloid leukemia. And I'm pleased to see that in your database, I played a little bit with the interface that you provided to us. This information is present. We were not able to conclude at the time because we couldn't find access to data sources with genomic information. Thank you.
Well, if I made, the FLT3 is included in the database. This is publicly available, the availability of the FLT3. You want to do a request, please send us an email and we will provide you the authorization to be in deep in the database trying to reply to your question. If you have a concrete question, we can reply in a period of a week. I have to tell you that there are no pediatric patients included in the database with genetic data because the information of AML in pediatric patient is very small one. But in adult patient is totally well recorded. FLT3 and FLT3 ratio also for over 8,500 cases around Europe, UK, Netherlands, Italy, France, Spain, and mostly Germany. Send us a request and we will reply you and we will reply for your question.
Yes, please.
Thank you. Here's a very nice presentation. I have one question. You mentioned you do an anonymization process of the data. So at which level patients have provided consent when you receive their data?
Okay, it was a long discussion because Harmony was born before the GDPR, so we have to be adapted to the GDPR. And we also have a bioethical advisory committee with 5 members. And we try to resolve the question in which should we go for 50,000 cases, most of them already passed away patients. So it is kind of material in which how we can manage this. We went to a legal bureau in Germany to obtain the way in which we can do this transfer of data. And the way in which we manage this is a double pseudonymization with a trusted third party. In the middle, that way, trusted third party is the Department of Epidemiology of the University of Ulm, Professor Gabriele Nagel, who is doing who is removing the label in order to don't re-identify patients. Any kind of identifier, and most of the quasi-identifier has removed it, and always the information is aggregated. And I also want to tell you that nobody can take the data, can bring the data to their computer. All the information and all the data that you are working on it, are you working inside the platform? The platform is now hosted in SINAF, which is institution very close to Bologna University in Italy.
Okay, thank you. Sabine, introduce the next speaker just before lunch break. Will be short.
Yes, short and sweet, but very relevant, because we will after lunch break, we will have breakout sessions, and we need a little bit of information on that. And it will be presented to you by Kelly. Kelly is a pharmacist by training, and she's very much involved in the real world evidence activities as part of the real world evidence workstream at the EMA Data Analytics and Methods Task Force. And she also coordinates the EMA HMA Patient Registry Initiative. So you're in very good hands. Please, Kelly, over to you.
Thank you, Sabine. And I will be very short because we are all hungry and want to go for lunch. Can I have the next slide, Valerie, please? Yeah. Thank you. You have seen this slide yesterday, presented by Carla. A big thank you to all of you who have responded to the survey sent to you a few weeks before the workshop. They have your responses have helped us a lot to prepare for the breakout sessions. 43 colleagues have responded, and you have here the breakdown by stakeholders type. Next slide, please, Valerie. And yeah, just in a nutshell to explain, so now we're going to break for lunch, and after lunch we directly go to our breakout sessions. In the handbooks you will see in which breakout you are, in which room. We will have 4 breakouts in parallel. So 2 of the groups will discuss how we can foster the use of patient registries for regulatory decision making through the data quality framework and its chapter, the catalogs, etc. And then 2 groups in parallel will discuss more in depth about the key elements for feasibility assessment to identify the relevant registries for specific research question. Next slide, Valerie, please. You can pass this one and the next, and then again the next. Yeah, thank you. So yeah, you will see it will be exactly like yesterday. You will have already. We have prepared some questions. So we have a plan. We will be back here in room 1C at 3:30 after the breakout and a little coffee. The same as yesterday, each group will have to appoint a rapporteur to report the outcome of the discussions from each group. And I think that's it. Just to clarify again that the breakout sessions are only for on-site participants, not for online participants. And I think I will stop here. Thank you very much.
Thank you. So it's time for lunch now. Lunch is just outside of the room. So we were able to only, I will say, not able to catch up with all the delays, but 5 minutes, 10 minutes, less than 10 minutes, it's fine. So we have 48 minutes because we really need to start sharp at 2 p.m. for the breakout session, and you need indeed to go in the different rooms. So please be on time and enjoy the lunch.
Okay, everybody, thank you. We're back. We're on the home straight, and we've had 4 busy and excited groups taking part in the discussion this afternoon. And hopefully we have 4 excited rapporteurs to tell us all about it.
Okay, then I'm going to talk a bit about our multi-stakeholder workshop, Group A. So next slide, please. So for our objectives, we focus on a data quality framework and the HMA catalog on data sources and interoperability between registries and data sources. So next slide, please. So we can, in our group, we all agreed that most in the room, we heard about this, and it is important to be considered. So part of the tools to be considered and the use of the registries for regulatory purposes. Also, we mentioned like other guidances we needed to use, which are like context of use, like registry-based study future reflection papers and interventional studies. Next slide, please. So what do we think about the draft DQF for European medicine and regulation? We all agree that it's a useful tool. It does explain like the what, but not exactly how. So the angle depends on this. Like the angle for this topic, we focus mostly on the regulatory aspect. Also, it is important to mention like for the how is more about the fundamental issues. So we came to a conclusion in the room like funding is still a concern which needs to be addressed. So also like how do we get people to collect the data also in a sustainable way? Of course, there is another issue which then arises like if you don't have enough money, like it's also then in return hard to obtain and maintain the quality of the data. So to do so, we need to reach out to everyone, think about like data holders, hospitals. So a question we got from the group is how can we make sure that it is applied? Next slide, please. So regarding if the document is clear enough, we would say like it should still need like concrete examples based on the context of use, like registry data. Think about like drug utilization, long-term efficacy, and safety studies. Also, another question which arises like there is clearly a need to identify strategies and data accuracy. There is a need to use data. It is necessarily and we need a proper approach to do so. Like think about the Bayesian or the real-world data. So the question we had, like, is it alignment of the guidance? Also, another aspect we concerned is about the involvement of the HTA. It is advisable to ensure that registry data can be used for drug reimbursement purposes. Yeah, next slide, please. So about the catalogs, we also agreed on that one, that it is a useful tool to use. Also, from the group, we realized like it is important to ensure that every country is aware of this existence of this catalog. So it's important to look at every country, how they are like in between promoting and to create awareness about this catalog existence. We need to ensure interoperability between the three platforms presented this morning. Another factor to consider is also the informed consent. So it is important to clarify to patients about this informed consent, as it will ultimately also have impact on the data quality. Next slide. Or is that it? Okay, then I think it's it. Thank you so much.
Thanks very much for a really clear outline of the discussion. And just for completeness, does anyone from the group want to add anything to that? Okay. If not, we'll move on to the next group that also considered the same question. Thank you.
Is this the slides? So we had a very fruitful and interesting discussion between academic, industry, registries, and regulators.
Sorry, just get the slides coming. Yes. Thank you. Yeah, and perhaps it's helpful, and I should have said it at the start, if you introduce yourself as the rapporteur. Thank you.
Okay. Yeah, my name is Annick Markslag. I'm from the registry from EBMT. As I said, we had a very interesting and fruitful discussion. So the data quality framework. Most people in the group had seen this. The document, were so aware. Was interesting that see that the patients are all aware. Can we go back to the first slide? Okay. Okay. Yeah. Okay. So there was But of course, people still, it's a new document to be implemented. So there's still people have to start working with it. The clarity aspects. Made some concepts of the parent document easier to grasp. Need to understand the whole chain of the data stream and responsibilities of each link. We had a very long discussion, for example, about data completeness. What is data completeness, and for what purposes? And so there's still some struggle to talk about data quality assessment without research questions. So, sort of a chicken and egg story. What do you want to collect to answer certain research questions, or do you collect data and then indeed you can do your studies based on the data that you collect? So the applicability potential of the chapter is appreciated, but some more experience is needed for implementation. Need for data source engagements, so physicians and patients. So it's very important that both physicians but also patients get feedback for the data that is collected. So I had one example in Sweden where investigators could immediately see how their patients were doing, and that was very promotive to indeed enter information into that database. So that's very important for people to understand that you don't only enter information in the database, but you need to get the data out. And the sooner and the more clear you get the data out, the more useful it is for people, and of course, most motivating it is to enter data into a database. Next slide. So then about the catalog. As with the framework, people had seen this before, is aware that it's implemented. It's considered to be useful in the landscape sense to what data exist rather than to start from zero. So very useful the search function within this tool. And it's seen as an improvement over the current PAS register. So important that we will see that it will grow over time. Still a limited number of registries are entered into the system. So we expect that over time it's more comprehensive. But some additional information would be valuable. For example, when it was the last update made, so that people know that the information that they are checking is indeed up to date and clear. So that's something that we advise to be added or to be included into this catalog. And a link to the data dictionaries. We've been discussing a lot about the dictionaries. It helps to develop the protocols. You need to know what kind of information, what kind of data is indeed collected. And we see that, at least that was the feedback, that often dictionaries are not shared, but it's very important to know how you collect data. For example, if you talk about age, is it certain groups, or is it the actual age? So that's something that you need to know to help develop the protocols. Next slide. And then we talked about the promotion of the registries. The ideas that the team came up with is about publication, key opinion leaders to have them involved. Also, letters published in journals about the registry, the protocol, what is collected. A suggestion about the registry village during a congress that was provided by our colleague from Sweden, who had a very good experience with the village. Academic industry forums, registry industry forums, also important that the EMA is included here, that they provide promotional campaigns and include information on the registries in their newsletters, and the registry network collaboration. I think that was it. Anything to add from the people that we discussed?
There's still one last slide.
Oh yeah, about the interoperability. Linked to other data sources, very useful. There are currently projects ongoing to investigate this. For some, it's the linkage between other data sources. It's by default. Also working with sister organizations, example outside of Europe. So very important to see how you can indeed work together with other registries and to collect data in a consistent way, so that information can be shared or can be compared. But of course, there are limitations with GDPR and national laws that might be stricter on patient consent. So we see that it's a general problem especially in different European countries, that data cannot be shared because patients have not provided informed consent to share the data. Questions about a federated model. A federated model is a model where data stays in the registry, but only the codes are shared. So the Darwin model to counter the GDPR limitation. So that is that might be an option, but the risk is to lose granularity with a common data model. So we know that implementing a new common data model for existing registries is very complex and difficult, but it could be a good advice when new registries start up that they get guidance on that common data model, and that it could be very useful to start with a new registry with that implemented. Thank you.
Thanks very much. So another broad-ranging and wise discussion, I think. And for completeness, anybody want to add anything to the discussion? Yes, thanks, Peter.
Just a question, because I was triggered by the village model at conferences. I like selling the ideas, what we're doing here, because there's still a very big need on expanding on what we are all trying to achieve, and we learn from each other. So could you elaborate a bit on that?
Yes, that was ECDRIMS. So ECDRIMS is the largest congress in multiple sclerosis. And for instance, 2 years ago, we had ECDRIMS here in Amsterdam, and then we had at the exhibition hall, we had a registry village with a couple of registries in the MS field. So we are like a big family there.
Peter's taken notes. Thank you. Thank you very much for that. And now let's move on to the second pair of groups. And they were discussing key elements of a feasibility assessment to evaluate the relevance of a registry in view of a specific study question and design, areas for collaborations to strengthen the use of registries for regulatory purposes and decision making. And for B1, we have a rapporteur, yeah?
Yep. Oh, I don't know. Are we B1 or 2?
You're one.
Okay. Thank you. I'm just waiting for the slides. Yeah, so we started out talking about fit for purpose and what it means. So first we defined that we're talking about disease registries, not product or treatment registries. So we focused just on disease registries. Fit for purpose for us means a registry is very broad. It is a resource that may answer many questions. It may be fit for purpose for some questions, but not other questions. So the question is the registry able to answer my research question or what I need to do with the registry? And if the answer is no, can we adapt it to be fit for purpose? One of the things we talked about was the design phase. When you're designing a registry, we should be thinking about what the data will be used for in 5, 10 plus years, not necessarily what it's used for right now. because things change and evolve very quickly. And everything around the registry and being fit for purpose. We talked about data quality, which is mandatory, so it doesn't fit into fit for purpose. It's kind of like a mandatory thing that has to happen for it to be fit for purpose. We need to understand the data collection process to have a full understanding of what data can be used for and what the limitations are. Then next slide, please. We talked about the data elements. So this is just to remind you what was in the survey. So the contexts of use. So the people who completed the survey prioritized these, and there's a list of several contexts of what the data can be used for, ranging from disease epidemiology, drug utilization, feasibility of a planned clinical trial, providing clinical trial arm, PMES, long-term follow-up, validate outcome measures, risk minimization measures, and data on patient experience. So this is just a reminder of what we were actually looking at. Next slide. So the question was, what are the key data elements to collect in a registry for the contexts that we just saw on the previous slide? So the core data set would include patient demographics, disease-specific data, treatment data, reason for treatment, regime, switching treatments, outcome data, meaning clinical outcome data, as well as patient-important data, including mortality data. Next slide, please. So that was the core. There were several other items that we discussed and considered. One is comorbidities and con-com medications. There's a challenge to collect these in registries because they can be very detailed and they change frequently. There was an option of linking to other data sources to get this type of data. We talked about if a treatment cures a patient, what will the follow-up of that patient be? If they no longer need to go to a treatment center, will they be lost to follow-up? Are there ways to maintain a data collection relationship with that patient? The time element is important when collecting data. So is the data being collected over time in a systematic way, and when is it being collected? Registries should be evolved as standard of care evolves. So particularly around outcomes, important outcomes may change over time as the science progresses. So if you start your registry looking at one specific outcome, you may want to adapt that outcome or adapt your registry to change that outcome. So you can add it, but you also need to think about stopping to collect other outcomes. Otherwise, your core data set is going to grow over time. And concentrate on what you really need so that you don't end up with a huge core data set. Next slide, please. So we then talked about key elements to consider when assessing the suitability or the relevance of a registry to address a research question. This is what you're seeing here is the response from the survey, and you can see the top 4. So the key elements for relevance include availability of key data variables, data quality, representativeness, and then it goes down to several other items. We in general agreed with this priority list. There was a couple that we thought, although important, they may not be as important for the relevance of a registry. So that included data collection and management processes and data sharing arrangements. Next slide, please. So this topic is about patient experience. Do you think patient experience data can contribute to regulatory assessment and decision making? So we had a big discussion about patient experience, the first thing was to define what patient experience is. There's no clear definition, so we need to come up with a definition and train people on what this actually means. Currently, PROs, or patient-reported outcomes, is a mixed bag. It includes quality of life data, patient-relevant data, psychosocial, psychological data. So there's a whole bunch of different types of data kind of put under the umbrella of PROs, or patient experience. So we need to tease that out. And they need to be contextualized. So the PRO that you use in your registry should be somehow contextualized to the disease state or the drug state. One of the members of our group gave an experience that she had. So she has 2 kids that have gone through 4 clinical trials. And from her experience, sometimes they're asked very odd questions. The PROs can be a burden on the patient, and they can lead to a loss of trust with the clinician. As an example, one of the questions that they had in their PRO was participating in this clinical trial better than a vacation? So she thought that was a little bit odd, and it was irrelevant and led to somebody not really wanting to go back to the clinic. And next slide is our last slide. Key areas where collaboration between stakeholders should be increased to strengthen the use of registries for regulatory purposes and decision-making. So the first thing, the planning of the registry should include all stakeholders right from the very beginning. This includes academia, clinicians, patients, industry, and regulators. We should pay attention to the registry patients who enter a clinical trial. So if a patient is in a registry and enters a clinical trial, they're then lost to the registry for that period of time because they can't participate in both. So it's something to be considered and understood a bit better, and maybe attempts to try and get them to be in the registry and the clinical trial at the same time. And registries, so registries should not come directly for qualification. It would be good if they went for advice in the beginning, either scientific advice or qualification advice, learned from that experience, and then came back for the full qualification. Thank you.
Thanks very much. A very broad-ranging discussion there, obviously. Anybody from the group like to add anything? Thank you. Yeah.
So I would just like to comment on the second point, because there was a lot of offline discussions after this. It was one of those things that was directed at industry, and even industry was rather confused about this because especially when it comes to rare diseases, it's not usual for patients that are participating in a trial. Imagine if it's a 3-year trial versus a 10-year trial, and if that patient participates in the trial but could not participate in the registry, then it's a massive loss for a registry. So the consensus is, or at least the advice that was given to the registry is, please find out which function from industry is barring this, and what are the reasons for this? Thank you.
And somebody at the bottom, yep.
Yeah, I'm also puzzled about that bullet point. I understand a patient cannot be in 2 clinical trials, but I didn't know a patient who is in a trial is out of a registry. And I would like to understand under which regulation is that the case? I guess essentially I'm challenging that.
I'd like to comment on this because in our registry, in Enroll, we keep people in Enroll. We have 2 modalities. When a company comes to us and wants to use patients from Enroll in their own trials, we told them that our preference is to keep the patient in enroll and make the adjustments, because occasionally there are some adjustments, like if there is the same cognitive assessment, if people do this twice, it can bring some bias to the analysis in the trial. So there are some things that need to be attended to. But we always tell companies that we prefer to keep the patient in the registry and in the trial. Fortunately, we have several of these cases. We have also a couple of companies that refuse completely, and what they offered was to give the data after the trial to fill the hole in the registry. And so we did that too. Happy to do more details outside.
But maybe what's important indeed, as put in the chat, I think, is that this information is recorded in the registry, so that you know that during that period, indeed, the patient was contributing to a specific clinical trial, which may explain that indeed the assessment is done on a different way than as per routine clinical practice. But yeah, excluding seems a bit weird indeed. Yes, please.
Yes, for us the same. We collect the data of patients who have been in a compassionate use program or in a clinical trial. It's depending on the selection you make. So if it's about registry patients, you do not select those patients in your selection, but there are in the registry. So we keep following them. But it's depending on the study that you do, what kind of patients you need for the study.
Yes, and I think this will be a critical question. So I'm looking at one row in the back, because indeed, if we want to use also the registries for randomized clinical trial, we need to ensure that you still have that opportunity.
Important point coming out of the discussions. Okay, if there's no further comment there, we'll move on to the second group who also discussed this question. Thank you. Who is the rapporteur? Cecilia, thank you. Cecilia.
So I'll do my best to summarize, and also in the interest of time, I'm not sure I will go through all the slides we have, and I'll summarize the discussion, and the other experts in the room can add to that. So we discussed the registries in several contexts and for 5 main purposes. So first, in the general case situation, what would be the core data set that you need to look at regardless of the context of use of the registries. Then for having registries for the purpose of epidemiology study or natural history studies, drug utilization patterns, safety studies, and then for the last one, we pick external control because we thought, okay, that's something that is also a hot topic, particularly in the context of rare disease and ultra rare disease. And I think that there is the need. The conclusion was that, okay, maybe it's very challenging to have a one-size-fits-all and anticipate what is going to be the use for each registries and different regulatory research questions. But definitely there is an opportunity to improve guidance and help to engage and communicate with external stakeholders that needs to be involved in the design of registries, but also the maintenance, and have the link between the impact of the registry on the regulatory decision making. And for example, a registry that is used for the purpose of simply informing a regulatory decision is not going to have the same impact in the regulatory decision making as a registry that is going to be used to generate new data, or even is going to replace data from control trials. And according to this type of framework that allow to link the registry, the use of the registry with the impact on the regulatory decision making, that would allow for external stakeholders, registry holders, to understand and engage early with the regulators to discuss the design of the registry. Maybe for those who have high impact on the regulatory decision making, have an enhanced pre-submission meeting and have a broad access to enhanced regulatory procedure, and have these registries built with key identified regulatory milestones.
One other important that was made is that the registry should be designed with having a broad spectrum of stakeholders. So the regulatory decision making is important, but also HTA and payers research question should be kept in mind when the registry is being designed in order to avoid to have a repetitive exercise that is being conducted and reduce the burden on all the different stakeholders involved in the design of the registries. I think that that's how I can summarize the discussion, and we have suggestions, of course, that are detailed in the slides, but in the interest of time, we will not go through those. So I don't know if Peter or Sabine or other experts who were in the room want to add to that. Thank you.
Thank you from my side. You did an excellent job. Thank you so much.
And we're doing all right on time if anybody else wants to pop in. Okay. So if not, what I can see, what we can both hear is such a broad discussion that was held across both groups this afternoon. And what's interesting is that even though we had duplicates, shall we say, in terms of topics with the two groups, that actually there's a huge amount of things that were individual to each group and things that complemented across the groups. And that's actually really valuable for the write-ups that will happen next and the reports and the activities that are going to grow from this, because it's not been a talk shop. What struck me listening to this is that much of the ongoing work is welcome, but people have made lots of suggestions to build on it. And especially there's a welcome for the data quality framework, including especially indeed the real world data and evidence chapter is particularly useful for people. And the other thing that's welcomed greatly is the HMA EMA catalogue, and the further interoperability of the different platforms is something I think that's anticipated and will be greatly welcomed, especially by multiple registry holders. We've had some useful suggestions that may go on hold until version 2 of the catalogue, given that it's going live on Thursday. But I think they've been well heard, and they'll be probably integrated in the future. And a really nice section that grew out of the promotion of registries and raising awareness about them to all sorts of groups, be it the users, be it patients that might want to contribute, be it people that are interested or need to conduct studies, or that are looking for data for whatever purpose. So that raising of awareness is crucial, and there were some really good ideas there. And then coming to the second group, certainly the concept of fit for purpose has gathered quite a lot of consideration, and I know a lot of that will be captured and will go into the report later, but a lot of thinking to be done around there. And one thing that struck me particularly from the conversations was the idea of the long 10-year time frame, looking forward not for what we need right this minute, but where we're going in the future, really important for future proofing. And then the huge and important issue around hearing exactly what's important out of the patient experience, and that being relevant in the context of whatever it is that's being studied, whatever drug, whatever disorder. And that is something I think that resonates with many people, both evaluating data and should resonate for planning studies for the future. Nice to hear the core data elements are pretty much highly recognized, and the value of ancillary information and core data elements strike me as so consistent with what we've been through in multiple registries before, and that is an important point going forward. So thank you for all of that. I will hand over now to Patrice to complete considerations. Thank you, Patrice.
Thank you, Patricia. So I will not take too much of time. I think I'm supposed to give the final words. So it has been quite extensive days. The last 2 days almost have enlightened the important role of patient registries in the development and monitoring of medicines to ensure that these are effective and safe for patients. And if we are all here today, it's because we are all convinced by this. And I think that over these 1.5 days, in fact, to be correct, we have collected a lot of valuable input from all of you, including on the EMA qualification of patient registries as discussed yesterday, on the data quality framework chapter, on real-world data sources, and on the steps to follow for the feasibility assessment of registry-based studies as discussed today. I will not go over all the details as you did it very well, Patricia, but otherwise you will miss your train and flight because indeed there were quite a lot coming out of these discussions. But I want also to reassure you that all this input will be included in the workshop report that will be shared for comments with the workshop participants and translated into recommendation, hopefully on actions and guidance needed to enable the use of registries for regulatory purpose, but also if possible, and that's also something that we heard today, even if we didn't discuss too much, for HTA purpose. All the presentation will be soon posted on the workshop web page of the EMA website, followed in the next weeks by the recording of today's plenary sessions and the report after consultation, as I said. I'd like to say a big thank you for your useful contributions. We look forward to further collaborating on the patient registry activities in the near future, and sure we will have additional opportunities for interaction on this topic. And I would like also to give a special thanks to all the chairs of the different sessions, to the facilitators of the breakout sessions, and to the team that organized the workshop, and especially Kelly Plushke, Carla Jonker, Valerie Muldom, that you don't see there, Juliana Vogt and Hannah Kankarnat. Wishing you a safe way back home.